In the stillness of night, when the world slows and thoughts begin to drift, there is another rhythm continuing quietly within you. Beneath the surface of skin, beyond the reach of conscious will, billions of tiny reactions hum softly in synchrony. Each one is a spark, a whisper of life, a moment where chemistry becomes vitality. This is the gentle pulse of cellular energy, the unseen current that sustains every heartbeat, every breath, every quiet thought that moves through the mind. Every living thing depends on this flow of energy. A single-celled bacterium drifting through water, a leaf turning towards sunlight, or a human resting in sleep. All are united by their need to capture, transform, and use energy. It is a universal requirement, not for motion alone, but for existence itself. Cells must constantly renew their components, repair their membranes, copy their DNA, and maintain the delicate balance of ions across their surfaces. None of these feats can occur without energy. It is as though each cell is a small lantern, fueled by a continuous flame that must never go out. If you imagine the body as a great city, energy would be its power grid, invisible but essential. Every structure, every process, from the flicker of thought in the brain to the contraction of muscle fibers, draws upon it. The energy itself is not visible, nor is it something you can feel directly. Yet, in the quiet hum of your organs, in the steady rhythm of your heart, its presence is constant and reassuring. The origin of this energy is ancient. Long before complex life took shape, the earliest microorganisms faced the same challenge: how to capture and store energy from their environment. Some absorbed energy from sunlight, while others drew it from simple chemical reactions deep within the Earth's oceans. Through countless generations, these primitive cells refined their methods, developing intricate pathways to collect and manage energy efficiently. Every cell in your body is a descendant of that ancient ingenuity. Each one carries within it a complete set of molecular tools for handling energy, honed by nearly 4 billion years of evolution. Though invisible to the naked eye, these processes are continuous and precise. The moment energy production falters, even briefly, life itself begins to dim. When scientists speak of energy within living systems, they are referring to a specific kind, not light, not heat, but biochemical energy, stored in the bonds of molecules. These bonds are like tiny coiled springs, holding potential power that can be released with delicate precision. When the cell needs to move, divide, or communicate, it does so by tapping into this molecular reservoir. It is a quiet exchange, a constant conversion between forms of energy that allows everything else to unfold. To appreciate the depth of this process, it helps to think of the scale at which it operates. Within each of your cells are countless molecular machines, enzymes, pumps, and motors, each performing their duties with tireless reliability. Some move ions across membranes, maintaining balance between the inside and outside of the cell. Others assemble proteins, stitching together amino acids in the correct sequence. Still others copy strands of DNA, ensuring that genetic information passes faithfully from one generation of cells to the next. Every one of these actions, from the smallest adjustment to the largest synthesis, depends on the steady supply of biochemical energy. The molecule that makes this possible is known as adenosine triphosphate, or ATP. Though we will explore its structure and function more deeply later, for now it can be thought of as the common currency of energy in biology. Just as a city might rely on electricity to power its lights, motors, and communication systems, cells rely on ATP to energize their work. It is produced constantly and used immediately, a molecular coin passed from one reaction to another. Without it, even the most fundamental cellular activities would come to a standstill. At any given moment, the human body contains only a small reserve of ATP, perhaps enough to last a few seconds if production ceased. This scarcity underscores the remarkable pace at which cells regenerate their energy supply. Within every second of your life, trillions of ATP molecules are being created, used, and replaced again. The process is ceaseless, even during rest, even in sleep. There is never a pause. Energy, however, is never created from nothing. It must come from somewhere. For living things, that source is food, the chemical energy stored in the bonds of sugars, fats, and proteins. These molecules, produced originally by plants through photosynthesis, are our link to the sun's energy. When you eat, you are not merely consuming matter. You are taking in the stored sunlight of the world, packaged by plants and delivered through the intricate web of life. Inside your cells, that captured energy is released in small, controlled steps, transformed into the form cells can use ATP. This conversion is carried out through pathways as elegant as they are complex. Each step is guided by enzymes, which act as molecular caretakers, ensuring that every reaction proceeds in order and at the proper pace. These pathways, glycolysis, the Krebs cycle, and oxidative phosphorylation are the foundations of cellular respiration, the grand process through which energy flows from food into usable form. Although they involve a cascade of chemical events, the underlying purpose remains simple: to extract the energy stored within organic molecules and make it available for life's work. Yet, this is not merely a story of chemistry. It is a story of balance. They must maintain harmony among competing demands: energy for motion, for repair, for communication. Too little energy, and essential systems begin to fail. Too much, and delicate molecular structures may become overwhelmed. The cell, in its wisdom, keeps all of this in equilibrium through intricate feedback systems, adjusting its pace like a musician keeping perfect time. It is a quiet process, but not a simple one. Inside every heartbeat, a vast symphony of reactions unfolds, from the splitting of glucose molecules to the pumping of ions across membranes. The mitochondria, those tiny, bean-shaped structures nestled within the cell, are its main concert halls. Here, electrons flow through intricate chains, gradients build and dissipate, and ATP is spun into existence with each rotation of a molecular turbine. The soundless activity of these organelles sustains every movement you make, every breath you take, and every dream that passes through your sleeping mind. If one were to look closer still, beyond the organelles and into the realm of atoms, the picture would remain the same: energy in constant motion, never lost, only changing form. It is the fundamental truth of physics and of life. Energy flows, transforms, and continues. A molecule of sugar becomes ATP. ATP powers muscle contraction. Contraction moves the body, and the energy disperses again, released as warmth into the air. In this way, the energy that animates you is never truly gone. It becomes part of the surrounding world, part of the quiet cycle of exchange that unites all living things. The biochemistry of energy, then, is not merely an academic subject. It is the hidden narrative of every moment you live. They continue their subtle labor, generating and using energy to preserve the fragile order that is life. Somewhere, deep within, a mitochondrion releases a molecule of ATP. A pump moves an ion across a membrane. A muscle fiber adjusts its tension slightly, even in sleep. To understand this is to glimpse something profound about existence. Life is not a single event, but a continuous series of transformations, a flow sustained by energy captured, stored, and released in delicate succession. That flow began in the first cells that ever lived and has continued unbroken through every lineage since. In your body tonight. As we drift deeper into this journey, we will explore how that current is created and maintained, how glucose becomes power, how the cell conserves its resources, and how all of life depends on the fragile dance of molecules that never truly sleep. But for now, it is enough simply to rest in the knowledge that energy moves through you, like a silent tide, constant and sustaining, the quiet fire that makes life possible. 2. What energy means in biology. When we speak of energy, the mind often turns to images of motion, a river flowing, a spark flashing, a flame burning softly in the dark. But in the realm of biology, energy takes on a quieter, more intricate form. It is not a force that shouts or blazes, but one that murmurs within every cell, guiding the delicate movements that sustain life. Biological energy is not something the body creates from nothing. It is something transformed, passed from one form to another, conserved through endless cycles of change. Energy, in its essence, is the capacity to do work, the ability to cause change, to move matter, to rearrange the patterns of atoms that make up the living world. It is the silent companion of all existence, flowing invisibly through every process that defines what it means to be alive. In human terms, we might say energy is what allows a thought to form, a muscle to contract, or a cell to divide. But at the molecular level, energy manifests in the most subtle exchanges, a phosphate group breaking free from ATP. These are the true heartbeats of biochemistry. Each one, a moment of transformation. The cells of living organisms perform countless tasks, and every one of them requires energy. They build complex molecules from simple ones, a process known as anabolism. They also break larger molecules down to extract energy in what we call catabolism. Together, these two sides of metabolism form the great balance of life: construction and destruction, synthesis and degradation, creation and release. The cell must weave these opposing processes into harmony, producing just enough energy to meet its needs without excess, storing some in chemical bonds and spending the rest to sustain its functions. Unlike machines built by humans, living systems are not powered by electricity or mechanical fuel. Instead, their energy lies in chemical bonds, the small but potent forces that hold atoms together in molecules. When these bonds are rearranged, energy can be released or absorbed. The cell captures this energy not in flashes or explosions, but in measured steps through reactions guided by enzymes that ensure every change occurs with precision and care. The result is a slow, continuous stream of usable power, never abrupt, never wasteful. At the core of this exchange lies a distinction between two main forms of energy: potential and kinetic. Potential energy is stored energy, poised for release, the tension in a stretched bow, the height of water behind a dam, or, within the cell, the bond between two atoms that could be broken to release energy. Kinetic energy, in contrast, is energy in motion, the flow of electrons, the rotation of molecular motors, the diffusion of ions through membranes. In living systems, potential energy within molecules is continually transformed into kinetic energy as cells perform their work, and kinetic energy is, in turn, captured again in chemical bonds to be used later. This exchange between stored and active forms of energy follows the same physical laws that govern the rest of the universe. The first law of thermodynamics, the law of energy conservation, tells us that energy cannot be created or destroyed, only transformed. For living organisms, this means that the energy they use must come from somewhere else. Ultimately, for most of life on Earth, that source is the sun. Plants, algae, and certain bacteria capture sunlight and convert it into chemical energy through photosynthesis. This energy, stored in carbohydrates and other organic molecules, when animals eat plants, or when one organism consumes another, that stored solar energy is transferred again, broken down, repackaged, and reused in new forms. Even as you rest, the warmth that radiates from your body is the distant echo of sunlight, transformed through countless intermediaries into the gentle heat of living tissue. The second law of thermodynamics adds another layer to this understanding. It states that in every energy transformation, Some energy becomes less ordered, typically dissipating as heat. In simple terms, this means that no process is perfectly efficient. Energy, as it flows, tends toward disorder. In biological terms, this is the reason living organisms must continuously take in energy from their surroundings. The body cannot simply reuse the same energy forever. Over time, energy spreads out, and its ability to perform work diminishes. To maintain their structure and function, living things must draw in new energy to replace what is lost to entropy. This is perhaps the most profound insight into why life is such a rare and delicate phenomenon. Living systems defy disorder, but only temporarily. They maintain internal order, structure, balance, coordination, by constantly exchanging matter and energy with their environment. Every breath, every heartbeat, every quiet pulse of ATP synthesis represents a momentary stand against the universal tide of entropy. Life is, in essence, the local reversal of chaos, achieved through the steady flow of energy. Cells use this energy to perform three main kinds of work. The first is mechanical work, the movement of muscles, the beating of cilia, the contraction of fibers that propel motion at every scale, from the flick of a flagellum to the movement of a human limb. The second is transport work, the movement of molecules and ions across membranes, maintaining the internal environment of the cell distinct from the world outside. This allows nerve impulses to travel, nutrients to enter, and waste to leave. The third is chemical work, the synthesis of new molecules, from DNA and proteins to hormones and structural components. Each of these types of work depends on energy being available in precise quantities at precise times. To manage this, cells have evolved a remarkable economy of energy flow. They do not let energy run freely. They channel it through carriers, molecules designed to hold and transfer energy in small, usable amounts. Chief among these is ATP, adenosine triphosphate, a molecule composed of adenine, a sugar called ribose. The bonds between these phosphate groups, particularly the last two, are rich with potential energy. When one bond is broken, the molecule becomes adenosine diphosphate, ADP, and a free phosphate, releasing a small burst of energy that can power nearly any cellular task. This transformation is reversible. Using energy from nutrients, cells can reattach the phosphate, recreating ATP and completing the cycle. In this way, ATP acts as a bridge between the energy released from food and the energy required for life's activities. It is both a storehouse and a messenger, a temporary form in which energy is captured, carried, and spent. Because ATP is used so rapidly, cells must produce it constantly. The human body, for instance, recycles its entire supply of ATP every minute, producing and consuming its own weight in this molecule each day. Yet the cycle remains balanced, the flow steady. On a deeper level, energy in biology is not merely about chemical reactions. It is about the preservation of structure against decay. Each cell exists in a delicate state of organization, with countless molecules arranged just so. To maintain this order requires constant input of energy, not dramatic bursts, but a steady trickle. It is what allows membranes to stay intact, proteins to fold properly, and DNA to replicate with precision. Without energy, these systems begin to unravel. A cell deprived of energy soon loses its structure, its chemical gradients collapse, and its contents drift toward equilibrium with their surroundings. Life ends not with violence, but with quiet stillness, the fading of organized motion into calm disorder. To think of energy in this way is to see life as a conversation between order and entropy, between the building up and the breaking down. Each organism, from a single bacterium to a human being, is a temporary island of order maintained by energy's flow. The warmth of your body, the electrical signals that pass through your nerves, the slow repair of tissue after injury, all are evidence of that exchange continuing quietly beneath awareness. Energy enters, performs its work, and leaves again, transformed but not lost. Even thought itself is powered by these same reactions. The brain, though it represents only a small fraction of the body's mass, consumes a vast share of its energy, a reminder that even the most intangible aspects of human experience are grounded in biochemistry. The dream, the memory, the slow drift of imagination before sleep, all are underwritten by the patient work of molecules transforming energy again and again, without rest. In the quiet symphony of life, energy is both the melody and the rhythm. It does not announce itself, yet it is everywhere, in every movement and every thought. Understanding what energy means in biology is to recognize that life is not a static condition, but a continuous process of transformation, of capturing, channeling, and spending energy in ways that preserve complexity for a little while longer. To live is to borrow order from the universe, to hold it briefly, and to pass it on. And so, as you rest and breathe, the gentle flow continues within you. Each cell carries out its work in silence, drawing from the energy of food, sunlight, and air, and returning it to the world in the soft warmth of life. Energy in biology is not just a concept, it is the quiet thread that connects all things, weaving motion, thought, and existence into one enduring tapestry of transformation. 3. ATP, the universal energy currency. Deep within every living cell, there exists a molecule so essential, so constantly in motion, that life itself depends on its continuous renewal. It is small, almost inconceivably so, and yet it holds the power to drive every heartbeat, every thought, every quiet cellular motion that sustains existence. This molecule is adenosine triphosphate, more gently known as ATP, and it is the universal currency of energy in the biological world. To understand ATP is to understand how life spends and saves its power. Just as societies use money to exchange goods and services, cells use ATP to exchange energy between chemical reactions. In this silent economy of life, ATP acts as both a coin and a key, unlocking the potential within other molecules and paying for the work that sustains the body. Without it, nothing moves, nothing grows, nothing continues. The structure of ATP is simple but profound. At its heart lies adenosine, a compound made of two smaller parts. Attached to this foundation are three phosphate groups arranged in a delicate chain. The last two of these phosphate bonds are high-energy bonds, not because they are especially strong. They sit like a coiled spring, ready to release energy when broken. When one phosphate group is removed, ATP becomes ADP, or adenosine diphosphate, and energy is set free in a form that the cell can use instantly. Sometimes, another phosphate is removed, forming AMP, or adenosine monophosphate, releasing even more energy. These transformations happen constantly, endlessly, in every living organism. The magic of ATP lies not in its size, but in its ability to couple reactions to link the release of energy from one process to the demands of another. Most chemical reactions within a cell are not spontaneous. Left to themselves, they would require more energy than they release, and would never proceed. ATP bridges that gap. When it is hydrolyzed, meaning its terminal phosphate bond is broken by water, it releases just enough energy to push those reluctant reactions forward. It is this coupling, this seamless exchange between giving and receiving, that allows the cell to maintain life's intricate balance. If you could watch this process unfold, it would appear almost like a dance of molecules. A strand of muscle fiber shortens as ATP binds to a protein called myosin, causing it to change shape. The phosphate is released, and energy ripples through the fiber, creating movement. In another cell, an enzyme captures ATP and uses it to join two smaller molecules into a larger one, perhaps building a strand of DNA or forming a complex lipid for a membrane. Elsewhere, ATP powers the sodium-potassium pump, one of the cell's most vital molecular engines. This pump moves ions across the cell membrane, maintaining the electrical potential needed for nerve impulses and muscle contraction. Each of these actions, every pulse, every spark of thought costs the cell a few molecules of ATP, which it spends without hesitation. The use of ATP is both rapid and vast. In the human body, an estimated 75 kilograms of ATP are used and regenerated every day, far more than the total amount present at any one time. The reason this is possible is that ATP is not stored in large quantities. It is produced on demand. Mitochondria, the cell's powerhouses, create new ATP continuously through processes that will unfold in later chapters. Glycolysis, the citric acid cycle, and oxidative phosphorylation. Each molecule of glucose that enters these pathways can yield dozens of ATP molecules. What makes ATP so perfectly suited to its role is its balance between stability and reactivity. It is stable enough to persist long enough to be transported within the cell, but reactive enough to give up its phosphate easily when needed. This balance ensures that ATP acts as a reliable, immediate energy source, a currency that can be spent quickly but never wasted. The energy released during ATP hydrolysis is about 7 kilocalories per mole, a modest amount by chemical standards but perfectly tuned to biological needs. Too much energy would damage delicate molecular structures. Too little would be ineffective. In this precise moderation, nature shows its remarkable refinement. But ATP's story is not only about providing energy, it is also about connection. It is the molecular bridge that unites every living thing. The same ATP molecule that fuels your heartbeat could, in another form, across the vast diversity of life, from bacteria to humans. ATP is the shared medium through which energy moves. It links the smallest cell to the largest creature, weaving a continuous biochemical thread through the web of life. The production and consumption of ATP form a dynamic cycle that never truly pauses. Within the cytoplasm, enzymes add phosphate groups to ADP, recreating ATP in reactions powered by the breakdown of food molecules. Within mitochondria, a more sophisticated mechanism operates. Electrons flow through a series of protein complexes embedded in the inner membrane, creating a proton gradient that drives an enzyme called ATP synthase. This molecular machine spins like a tiny turbine, joining ADP and phosphate into new ATP. Every turn of this enzyme produces a fresh burst of energy, ready to be used in countless cellular reactions. Even within plant cells, which capture energy from sunlight, ATP plays the same role. In photosynthesis, light energy excites electrons within chlorophyll molecules. These high-energy electrons move through a similar transport chain, producing ATP that fuels the creation of sugars. Whether in the chloroplasts of plants or the mitochondria of animals, the logic remains unchanged. Energy is captured, stored, transferred, and used always in the currency of ATP. Despite its centrality, this constant turnover reflects both the energy demands of life and the efficiency of the systems that meet them. There is a quiet elegance in this process, a continuous renewal that mirrors the cycles found throughout nature. Just as water evaporates, condenses, and falls again as rain, so too does energy circulate endlessly within the living world, with ATP as its vessel. Beyond its role as an energy carrier, ATP also acts as a signaling molecule, a messenger between cells. In the nervous system, ATP can be released into the space between neurons, influencing communication and coordination. It helps regulate blood flow, immune responses, and even pain perception. In this sense, ATP is not merely a form of fuel, but a language of energy, a way for cells to speak to one another in the shared dialect of chemistry. The story of ATP also hints at life's deep unity with the physical laws of the universe. It demonstrates that even the smallest structures obey the same principles of energy conservation and transformation that shape galaxies and stars. The energy that drives a muscle contraction once powered the fusion of atoms in the heart of the sun. Through an unbroken chain of transformations, that same energy now flickers within you, organized by cells, directed by enzymes, made delicate and purposeful by the structure of ATP. It is almost poetic that such an essential molecule is also so ephemeral. It is not hoarded or accumulated, but created, used, and replaced in an endless cycle of giving and receiving. In this, ATP mirrors the rhythm of life itself, an ongoing exchange where energy flows freely but always returns in another form. The warmth of your hands, the motion of your lungs, the gentle beating of your heart, each of these arises from countless ATP molecules completing their brief, purposeful existence. When viewed with quiet attention, this process feels less like chemistry and more like a form of grace. There is harmony in the way the cell spends its energy, precision in the timing, and beauty in the balance. Nothing is wasted. Nothing is hurried. The same molecule that flickers into being within your cells tonight will be broken down and rebuilt countless times before morning. Through it, life maintains its steady rhythm, not with noise or spectacle, but with the silent, tireless pulse of molecular motion. And so, as we consider ATP, we glimpse not just a molecule, but a metaphor for existence itself. Life is an ongoing act of exchange, a continual giving and receiving of energy, structured and sustained by fragile bonds that hold just long enough to make meaning before they are released again. ATP is the quiet fire of being, the molecular signature of life's persistence. It reminds us that in every living thing, from the smallest cell to the beating human heart, energy is both a gift and a cycle, eternally flowing, eternally renewed. 4. The cycle of ATP and ADP. Within every cell, life unfolds in a series of patient, repeating rhythms. Some are visible to us: the rhythm of breathing, the pulse of the heart, the rise and fall of the chest as air moves through the lungs. Others are hidden, existing on scales so small that even the most powerful microscopes can only hint at their quiet motion. Among these invisible rhythms is one of the most essential of all: the continuous transformation between ATP and ADP, a molecular cycle that fuels the entirety of life. It is a process so constant that, even as you read these words, it repeats countless times within you each second. Without it, the body's stillness would not be peace, but silence, the silence of life, at rest forever. Adenosine triphosphate, or ATP, can be thought of as the body's charged battery, ready to release energy when a cell needs it. Its close partner, adenosine diphosphate, or ADP, is the discharged form waiting patiently to be recharged again. The transformation between these two states is one of nature's most elegant transactions. Each time a phosphate group is detached from ATP, a small but potent amount of energy is released. The molecule becomes ADP, carrying 2 phosphate groups instead of 3, and in the process, it powers one of the countless reactions that make life possible: the contraction of a muscle, the movement of ions, or the assembly of a protein. Then, as soon as energy is available again from food or sunlight, ADP is reunited with another phosphate group to form ATP once more. In this endless exchange, the beauty of this cycle lies in its simplicity. It is not a violent release of energy, nor a sudden burst of reaction, but a measured, reversible flow. ATP does not explode like a spark. It yields its energy gently, in amounts small enough for the cell to control. When it is used, ADP is not discarded but recycled, immediately ready to become ATP again. This closed-loop system allows cells to sustain energy production continuously without ever depleting their resources. In this way, ATP and ADP form a kind of molecular heartbeat. To appreciate the scope of this process, one might imagine the number of ATP molecules in a single human cell. At any given time, A cell contains only enough ATP to last a few seconds if production were to stop. Yet it never runs out, because the conversion between ATP and ADP is so fast, so efficient, that the balance is constantly maintained. Every second, billions of ATP molecules are being broken down into ADP, releasing energy for cellular work. At the same moment, new ATP molecules are being formed from ADP and inorganic phosphate, replenishing the supply. This is not a cycle that pauses or slows. It continues unbroken from the first moment of life until the last. The recharging of ADP into ATP is one of the most vital operations in all of biology, and it occurs through several interconnected pathways. In the cytoplasm, the simplest of these is glycolysis, a sequence of reactions that break down glucose molecules into smaller fragments. As the bonds within glucose are rearranged, some of their stored energy is used to attach phosphate groups to ADP, forming ATP directly. Though this process yields only a small amount of ATP, it represents one of the oldest and most universal energy pathways in existence, a method so ancient that even the simplest microorganisms rely on it. Within more complex cells, however, most ATP is generated in the mitochondria, those tiny, folded organelles, often called the powerhouses of the cell. Here, a far more elaborate process takes place. Nutrients from food, already partially broken down, enter a series of reactions known as the citric acid cycle or Krebs cycle. These reactions strip away electrons and hydrogen atoms from the fuel molecules, transferring them to carriers such as NADH and FADH2. These carriers then deliver their high-energy electrons to a chain of proteins embedded in the inner membrane of the mitochondrion. As the electrons move along this chain, they release energy that drives the pumping of hydrogen ions across the membrane, creating an electrochemical gradient, a kind of stored potential, much like water held behind a dam. It is this gradient that provides the power to regenerate ATP from ADP. The ions flow back through a remarkable molecular machine known as ATP synthase, which acts like a microscopic turbine. As protons pass through it, the enzyme spins, binding ADP and phosphate together to form new ATP molecules. This process, called oxidative phosphorylation, is astonishingly efficient. A single molecule of glucose, fully oxidized, can produce around 30 to 32 molecules of ATP. In the quiet darkness of each mitochondrion, thousands of these molecular turbines spin continuously, turning the potential energy of food into the chemical currency of life. The return of ADP to ATP through this process is more than a mechanical exchange. It is a restoration of readiness, a return to potential. The moment a cell uses ATP and converts it to ADP, it creates a demand that drives these energy-producing pathways into motion. The system regulates itself through feedback, ensuring that when energy levels fall, the rate of ATP synthesis rises to meet the need. When energy is abundant, production slows, conserving resources. In this way, the cell maintains a delicate equilibrium, always producing just enough energy to match its requirements. Even as ATP and ADP cycle endlessly, there is a third molecule that occasionally joins the pattern, AMP, or adenosine monophosphate. When energy demand is exceptionally high and ATP stores are depleted, ADP molecules can transfer a phosphate group between themselves, forming one molecule of ATP and one of AMP. This reaction serves as an emergency measure, a way for the cell to sustain energy flow even under stress. Enzymes within the cell monitor the relative concentrations of ATP, ADP, and AMP. When AMP levels rise, it is a clear message that energy reserves are low, and pathways like glycolysis accelerate to restore balance. In the larger sense, the ATP-ADP cycle, every synthesis of a molecule, every signal sent across a nerve, every movement of a muscle fiber depends upon it. The energy released as ATP becomes ADP is transformed into many forms: chemical, electrical, and mechanical, but its origin remains the same. It is the cell's way of spending energy, just as the reformation of ATP is the act of saving it again. This rhythmic exchange sustains not only individual cells, but entire organisms, from the smallest bacteria to the tallest tree. If one were to step back and imagine this process on a global scale, it would appear as an unbroken flow of energy linking all living things. The ATP produced in a leaf through photosynthesis might become the energy that fuels an animal that eats that leaf. The animal, in turn, breathes out carbon dioxide, which the plant will use again to make new sugars. Those sugars will later form new ATP molecules, completing the circle. In this way, the ATP-ADP cycle in your own cells is part of a much larger pattern, a continuous exchange of energy that connects every living organism on Earth. At night, as your body rests, the rhythm of this molecular exchange does not cease. Muscles repair themselves, neurons restore their delicate chemical balances, and each of your trillions of cells continues the quiet conversion of ADP to ATP and back again. Even in sleep, energy is spent and renewed, the flame of life never extinguished. The warmth of your body in the darkness, the steady beat of your heart, the slow rise and fall of your chest, all are the visible reflections of this invisible cycle, unfolding in perfect silence beneath your awareness. To think of the ATP-ADP cycle is to realize that life's continuity depends not on permanence, but on constant renewal. Nothing in a living system stays fixed. Every bond that breaks is reformed, every molecule replaced, every loss answered by creation. The same molecules of ATP that sustain you in this moment will soon be gone, replaced by others just like them, created from the energy of the food you have eaten or the oxygen you have breathed. In this unending exchange, life affirms itself, transforming, adapting, and renewing in each instant. The quiet power of this cycle lies in its humility. It is not grand, not dramatic, and yet it underlies everything that lives. In the simplest bacterium and the most complex human being, ATP and ADP continue their patient dance, one yielding, the other replenishing. In perfect balance, the process never stops, and because of that constancy, neither do we. This ceaseless exchange is the heartbeat of existence, a silent rhythm that began billions of years ago and still continues, steady and soft, within every cell that has ever lived. 5. Energy from food. The path begins with glucose. All life depends on energy. But before energy can be used, it must first be captured and stored in a form that cells can understand. For most living beings, that story begins with food, the physical expression of sunlight, transformed through the slow and intricate art of biology into something tangible. Whether it is a grain of rice, a slice of fruit, or the simplest drop of honey, every morsel carries within it a memory of light, bound in chemical form. In this sense, food is not merely nourishment, it is concentrated sunlight, stored in the delicate architecture of molecules. And at the heart of this process lies one simple sugar, glucose. Glucose is the quiet hero of cellular energy. It is a small, 6-carbon molecule, soluble, versatile, and rich in potential. Nearly every organism on Earth, from the smallest bacterium to the largest mammal, has evolved ways to use it. Within the body, glucose serves as the primary fuel, the substance from which most of the cell's energy is derived. It enters the bloodstream after meals, traveling gently through the body until it reaches cells that draw it inward like a tide. There, the real work begins: the stepwise liberation of its stored energy, guided by enzymes, transformed into the usable currency of life, ATP. The journey of glucose is one of gradual release, not explosion. Instead, cells extract this energy through a carefully regulated sequence of reactions, known collectively as cellular respiration. This process allows the cell to capture the energy contained within glucose molecule by molecule, atom by atom, transforming it into the precise amounts of ATP needed for life's tasks. The first of these stages unfolds in the cytoplasm and is called glycolysis. Glycolysis is an ancient pathway, older than oxygen itself. It takes place in almost every cell type and requires no specialized organelles or oxygen gas. It begins when a single molecule of glucose, composed of 6 carbon atoms, is primed by 2 small investments of ATP. This initial cost may seem counterintuitive, spending energy to gain energy, but it is necessary to make the glucose molecule more reactive, preparing it for the transformations to come. The phosphorylated glucose then rearranges into a new form and splits cleanly into 2 3-carbon fragments known as pyruvate. Along the way, 4 ATP molecules are produced, yielding a net gain of 2 ATP for each glucose consumed, along with 2 high-energy carriers, called NADH, that hold electrons for later use. Though modest in its yield, glycolysis is vital. It is fast, efficient, and reliable, allowing cells to extract a small but crucial portion of glucose's energy, even in the absence of oxygen. For single-celled organisms that live in low-oxygen environments, glycolysis may be the entire energy system. For more complex life, it serves as the first step in a longer, more elaborate journey that leads deep into the mitochondria, the cell's energy-producing centers. Once glycolysis has run its course, the newly formed pyruvate molecules must decide their fate. If oxygen is scarce, as it might be in a muscle pushed to exhaustion, pyruvate will enter an alternate route called fermentation. In human cells, this means it is converted into lactic acid, a temporary byproduct that allows ATP production to continue even without oxygen. When oxygen returns, the lactic acid can be recycled back into pyruvate. In yeast and certain bacteria, fermentation takes a different turn, producing ethanol and carbon dioxide instead. These humble chemical processes, refined over billions of years, are what give rise to the bubbling of bread dough and the fermentation of fruit into wine. In the presence of oxygen, however, the path of glucose deepens and expands. Pyruvate is transported into the mitochondrion, where it undergoes a brief transformation. Each molecule of pyruvate loses one of its 3 carbon atoms as carbon dioxide, a whisper of waste that will soon leave the body through exhalation. The remaining 2-carbon fragment combines with a molecule called coenzyme A to form acetyl-CoA, the key that opens the door to the next great cycle of energy extraction, the Krebs cycle. This transition step may seem small, but it marks the point where glucose, once whole, is fully committed to being dismantled for its stored energy. The Krebs cycle, also known as the citric acid cycle, is a series of reactions that occur in the fluid matrix of the mitochondrion. Each turn of this cycle begins when acetyl-CoA merges with a 4-carbon molecule called oxaloacetate to form a 6-carbon compound known as citrate, the same acid found in citrus fruits. Through a sequence of chemical reshufflings, this compound is gradually stripped of its carbon atoms, which leave as carbon dioxide. Each step releases energy, captured in 3 main forms: ATP, NADH, and FADH2. The NADH and FADH2 molecules are the true harvest of this stage high-energy carriers that will later donate their electrons to drive the production of vast amounts of ATP. By the end of the cycle, the original 4-carbon molecule is regenerated, ready to begin again with a new acetyl-CoA. From 1 molecule of glucose, 2 acetyl-CoA molecules are produced, meaning the Krebs cycle turns twice for every glucose processed. The result is a small amount of ATP directly produced and a much larger collection of reduced electron carriers, 12 in total, that contain the majority of the energy once held within glucose's bonds. But this energy still remains locked in potential form, waiting for its final transformation in the last and most intricate stage of respiration. This next phase, called oxidative phosphorylation, takes place along the inner membrane of the mitochondrion. It is here that the electrons carried by NADH and FADH2 are transferred through a series of protein complexes, collectively known as the electron transport chain. As the electrons move from one complex to the next, their energy is used to pump hydrogen ions across the membrane, creating a gradient, a quiet, invisible tension between two sides of the mitochondrial wall. It is this gradient that powers the enzyme ATP synthase, which spins gracefully like a molecular turbine, joining ADP and phosphate to form new ATP molecules. From the full oxidation of a single glucose molecule, this system can produce more than 30 ATP molecules, the richest yield in biology. Yet even this remarkable process is not entirely efficient. Some of the energy is lost as heat, contributing to the warmth of living bodies. This gentle warmth, often unnoticed, is a byproduct of life's inner fire. When you sit quietly in a dark room, the soft heat that radiates from your skin is the outward expression of millions of glucose molecules being oxidized within you, each one contributing its share of energy to sustain your heartbeat and your breath. The path from food to energy is, in truth, a cycle that never ends. The glucose in your blood may have come from a meal, but its energy originated in sunlight, captured by plants through photosynthesis. In that process, carbon dioxide and water are transformed into glucose and oxygen, the exact materials that your cells now use in reverse to generate energy. When you exhale, the carbon dioxide released is returned to the atmosphere, ready to be used again by another plant, somewhere else, at another time. In this way, the act of living connects you directly to the world around you, linking your breath to the wind, your warmth to the sun, and your existence to the great exchange of matter and energy that unites all life. Within every cell, glucose's journey from nutrient to energy mirrors the balance of giving and receiving that sustains existence. It begins as potential, a stored promise of sunlight, and ends as motion, heat, and renewal. The process never hurries, never pauses, but moves at the perfect pace to meet the body's needs. Through this steady rhythm, cells are kept alive, tissues are nourished, and the flame of life continues to burn, quiet and bright. And so, the next time you eat, whether a piece of fruit or a simple grain of bread, you might imagine the hidden story within it. Each bite contains the memory of sunlight, the work of leaves, the movement of water, and the silent labor of the earth. Within hours, that energy becomes part of you, transformed into motion and warmth, thought and breath. It is a reminder that every living thing is sustained by the same flow of energy, moving through different forms, yet always the same, in essence. The path of glucose, from food to ATP. 6. Glycolysis, the ancient pathway. Long before animals breathed oxygen, before green plants stretched toward sunlight, and before the world's air was even fit to sustain life as we know it, there existed a simple, silent process, a way for the earliest cells to harvest energy from their surroundings. It required no light, no oxygen, no complex organelles or delicate membranes. It happened quietly in the watery interiors of primitive cells, in the dim oceans of a young earth. That process is known as glycolysis, a word that means, quite simply, the splitting of sugar. It is one of the oldest biochemical pathways on the planet, and to this day, it continues unchanged within nearly every living cell, from the smallest bacterium to the most intricate organism. Glycolysis is the beginning of the cellular journey that turns food into energy. It is the first chapter in the story of respiration, where glucose, the 6-carbon sugar that fuels nearly all life, is gently dismantled to release the energy held within its chemical bonds. What makes glycolysis so remarkable is its universality. Every living cell, no matter how different its structure or function, depends on this same process. It is as if evolution, after discovering it billions of years ago, found it so perfect, so efficient, that there was never any need to change it. Inside each cell, glycolysis unfolds in the cytoplasm, the thick, gel-like substance that fills the space between the cell membrane and the organelles. There, enzymes act as careful guides, ensuring each chemical reaction occurs at precisely the right moment. These enzymes do not rush. They work in a calm and deliberate sequence, one step following another, transforming a single molecule of glucose into two smaller molecules called pyruvate. Along the way, energy is both invested and earned. Electrons are collected. It is a process both simple and profound, capable of sustaining life even in the absence of oxygen. The journey begins with a small investment. The cell uses two molecules of ATP, the very currency of energy it aims to produce to prime the glucose molecule. This initial expenditure may seem paradoxical, but it is an act of preparation, a necessary input that allows the sugar to be reshaped into a form more reactive and ready to yield energy later. Once activated, the glucose molecule is rearranged and then split in half, forming two 3-carbon compounds known as glyceraldehyde 3-phosphate. Each of these smaller molecules continues through a series of reactions that ultimately release more energy than was first invested, producing 4 ATP molecules and 2 high-energy electron carriers called NADH. In the end, glycolysis provides a net gain of 2 ATP molecules for every molecule of glucose that enters the pathway. It is not a large sum, a modest profit in the grand economy of energy, but it is enough to sustain the most basic forms of life and to provide the first spark of power that larger organisms can build upon. Perhaps even more importantly, glycolysis provides the intermediate products that link it to other energy pathways, such as the citric acid cycle and oxidative phosphorylation, where the bulk of the cell's energy will later be made. What makes glycolysis particularly beautiful is its independence. It requires no oxygen, no specialized structures, and no external light. It can occur in nearly any environment, from the deep ocean floor to the interior of human muscle cells during intense exertion. In this way, it is a process that unites all forms of life under a single principle. Energy can be extracted from the simplest of molecules, even in the most inhospitable of conditions. This ancient resilience is what allowed early life to flourish long before Earth's atmosphere was filled with oxygen, and it remains a lifeline for cells that must occasionally survive without it. When oxygen is plentiful, the pyruvate produced by glycolysis travels onward, entering the mitochondria to be further broken down in the citric acid cycle. But when oxygen is scarce, as in a muscle deprived of air during strenuous activity, another path opens: fermentation. Through fermentation, the cell can continue producing ATP by converting pyruvate into other substances, such as lactic acid in animals or ethanol and carbon dioxide in yeast. Though this route yields no additional energy beyond what glycolysis provides, it ensures that ATP production never completely stops. It is a remarkable adaptation, a molecular backup plan that allows life to persist even in hardship. This ability to operate with or without oxygen gives glycolysis a timeless quality. It connects modern cells to their most ancient ancestors, those first living organisms that inhabited a world without breathable air. The chemistry that sustains your cells as you read this, the quiet conversion of glucose to pyruvate, is the same chemistry that may have powered the earliest bacteria billions of years ago. Through uncounted generations, this process has endured, not as a fossil of the past, but as a living, breathing mechanism that continues to hum within us all. Glycolysis is more than a means of generating ATP. It is also a foundation for other metabolic processes. The intermediate compounds it produces can be diverted into various biochemical pathways to synthesize amino acids, lipids, and nucleotides, the essential building blocks of proteins, membranes, and DNA. In this sense, glycolysis is both a source of energy and a crossroads of creation, linking the processes that break molecules down with those that build them anew. The cell's metabolic web radiates outward from this central pathway, demonstrating the elegant efficiency of life's design. On a molecular scale, the energy released during glycolysis does not occur in violent bursts, but in controlled transitions. Enzymes carefully regulate each step, ensuring that the release of energy happens gradually and safely. This control allows the cell to capture as much usable energy as possible, rather than wasting it as heat. If one were to look closely enough, one could imagine a quiet procession of molecules exchanging electrons and rearranging bonds, each step dependent on the one before it, all occurring in perfect synchrony. It is not a mechanical process, but a graceful choreography, repeated endlessly in silence. In human life, glycolysis plays a special role during times of immediate need. When a person runs, lifts, or exerts themselves intensely, their muscles demand energy faster than oxygen can be delivered to every cell. In those fleeting moments, glycolysis steps forward as the rapid supplier of ATP, operating anaerobically to provide the fuel necessary for motion. Though this form of energy production is temporary and less efficient, it is essential for survival, allowing the body to act swiftly in moments of urgency. The soreness that sometimes follows the result of lactic acid accumulation is a gentle reminder of this ancient pathway still doing its work. Yet glycolysis is not only about the present moment, it is also about continuity. It links the immediate needs of the cell to the long cycles of evolution and adaptation. The fact that it exists in nearly every organism alive today is a testament to its perfection. Evolution, which endlessly refines and replaces, has left this process largely untouched. It is a universal inheritance, shared equally by all living things, a whisper from the dawn of life that still echoes through time. Even in its simplicity, glycolysis encapsulates the essence of biological balance, the interplay between investment and return, between effort and reward. The cell gives a little energy to unlock the greater potential within glucose, just as nature invests in growth to harvest abundance later. The pattern repeats itself across all scales of existence, from the metabolism of cells to the rhythms of ecosystems and the cycles of the planet itself. As you rest, billions of these reactions occur within you every second. Glucose molecules enter your cells, their bonds loosen and rearranged, their energy gently released and transformed into the quiet hum that keeps you alive. There is no noise, no light, no interruption, only the steady, ancient rhythm of glycolysis, a process older than air, yet still essential to the warmth and motion of life. If there is something deeply comforting in this thought, it is because glycolysis reminds us of our connection to the earliest forms of existence. Beneath the complexity of human biology lies the same fundamental chemistry that once stirred in the first living cell. It is a bridge through time, linking every breath and heartbeat to the world's beginnings. In the deep stillness of your body, this ancient pathway continues to flow, as it has for billions of years, unbroken, eternal, and quietly alive. 7. The transition to the mitochondrion. As the process of glycolysis draws to a close, two small molecules emerge from the cytoplasm's intricate web of reactions. They are pyruvate, the 3-carbon remnants of glucose, and they represent the first tangible harvest of energy extraction. But in their current form, they are only partly transformed. Much of glucose's original energy still lies locked within them, waiting for a deeper and more refined process to release it. To reach the full potential of that energy, the journey must continue inward, into one of the most remarkable structures ever to evolve within a living cell, the mitochondrion. The mitochondrion is often called the powerhouse of the cell, but this simple phrase conceals a far deeper and more fascinating truth. Each mitochondrion is like a small living system within another living system, complete with its own DNA, membranes, and enzymes. They float gently in the cytoplasm, numerous and tireless, like embers glowing within the shadowed interior of the cell. The number of mitochondria varies depending on the energy needs of the tissue. A single muscle cell might contain thousands, while simpler cells may need only a few. Together, they form a vast network of energy production, ensuring that every breath, every movement, and every quiet thought can be sustained without pause. Before the pyruvate molecules can enter this inner realm, they must pass through the outer mitochondrial membrane, a gate that allows small molecules to slip through but regulates larger or charged ones. Beyond this lies the inner membrane, folded into countless ridges known as cristae. These folds dramatically increase the surface area available for the energy-producing reactions that will soon unfold. It is here, deep within this folded architecture, that the most intricate stages of cellular respiration take place. But first, the pyruvate must undergo a transformation. This step, known as the link reaction or the transition step, is the bridge between glycolysis in the cytoplasm and the citric acid cycle within the mitochondrion. As pyruvate crosses into the mitochondrial matrix, the fluid-filled space at the organelle's core, it encounters an enzyme complex called pyruvate dehydrogenase. This complex acts like a molecular customs station, meticulously processing each incoming molecule before it can proceed. One carbon atom is removed from pyruvate as carbon dioxide, a waste product that will eventually leave the body through exhalation. The remaining 2-carbon fragment is attached to a small carrier molecule called coenzyme A, forming acetyl-CoA, the molecule that will enter the citric acid cycle. In this transition, energy is released, though not in the form of ATP. Instead, it is captured by another molecule, NAD-, which accepts high-energy electrons to become NADH. This NADH will later carry its electrons to the inner mitochondrial membrane, where they will join the electron transport chain, the grand finale of cellular energy production. Thus, even in this small preparatory step, the groundwork is laid for the far greater transformations to come. Acetyl-CoA, now ready, is a pivotal molecule in metabolism. It is more than just a participant in the citric acid cycle. It is a crossroads molecule, connecting the breakdown of carbohydrates, fats, and proteins into a unified network of energy metabolism. Whether the cell is using glucose from food, fatty acids from stored lipids, or amino acids from proteins, all roads converge at acetyl-CoA. It is the universal entry ticket to the mitochondrian's inner workings, the molecule through which the energy of life is refined and released. The transition from glycolysis to mitochondrial respiration also marks a shift in the cell's relationship with oxygen. Glycolysis, as ancient as it is, functions perfectly well without it, yielding only small amounts of ATP. But the mitochondrian's machinery depends on oxygen's presence. Here, oxygen acts not as fuel, but as the final acceptor of electrons, the quiet anchor that ensures the entire chain of reactions continues smoothly. Without oxygen, the electron transport system would grind to a halt, electrons would have nowhere to go, and energy production would collapse. This is why oxygen, invisible yet vital, is the silent partner of all complex life. The structure of the mitochondrion reflects the precision of this process. The outer membrane is smooth and permeable, while the inner membrane is selectively permeable and intricately folded. The folds, or cristae, are more than mere structural features. They provide a vast surface area upon which the critical enzymes and transport proteins of respiration are embedded. Within this labyrinth of folds, electrons flow, protons are pumped, and ATP is synthesized in a continuous, balanced rhythm. The space between the two membranes, known as the intermembrane space, becomes a temporary reservoir for protons, storing potential energy that will later be used to drive ATP synthesis. Inside the inner membrane lies the matrix, a calm, concentrated environment where the citric acid cycle takes place. Here, acetyl-CoA enters a series of enzyme-catalyzed reactions that complete the oxidation of what began as glucose. Carbon atoms are released as carbon dioxide, hydrogen atoms are carried away by NADH and FADH2, and a small amount of ATP is produced directly. But the true energy harvest remains in the electrons carried by NADH and FADH2, which will soon enter the electron transport chain. It is in this transition to the mitochondrion that we can begin to appreciate the remarkable efficiency of life's design. Nothing is wasted. Each step flows naturally into the next, each molecule handed off to another in a seamless exchange. The energy extracted from food in glycolysis is not simply lost as heat or light, but is conserved, repackaged, and transferred into new molecular forms that can be used again. The mitochondrion acts as the cell's grand refiner, taking in the raw products of earlier reactions and turning them into pure, usable energy in the form of ATP. From an evolutionary perspective, the mitochondrion carries with it an extraordinary history. Scientists believe that these organelles were once independent bacteria, living freely in the ancient oceans. Long ago, a larger cell engulfed one of these bacteria, not to consume it, but to form a partnership. The smaller organism provided energy in exchange for shelter and nutrients, and over millions of years, the relationship deepened into interdependence. The once free bacteria became the mitochondria we know today. Their autonomy surrendered, but their purpose enhanced. This ancient alliance, known as endosymbiosis, remains one of the most significant events in the history of life. It gave rise to complex cells, eukaryotes, and made possible the evolution of multicellular life. Every mitochondrion within your body still bears traces of that ancestry. It contains its own small ring of DNA, separate from the DNA in the nucleus. It replicates independently, dividing and multiplying as the cell's energy needs grow. When viewed under a microscope, a mitochondrion even resembles its bacterial ancestors, a living reminder that cooperation, as much as competition, has shaped the course of evolution. Each one of these tiny structures within you is a remnant of an ancient partnership, still fulfilling its role billions of years later. The transition of pyruvate into the mitochondrion is more than a passage between cellular compartments. It is a step into the heart of life's energy economy. Here, in this quiet organelle, the cell learns to refine its resources, to draw from the simplest of molecules a nearly endless supply of power. The energy that began as sunlight, stored in glucose, will soon be transformed into ATP, the molecular coin that drives everything from muscle contraction to memory formation. As you breathe, oxygen enters your lungs and passes into your bloodstream. It travels silently through your body, reaching every cell, every mitochondrion. Each oxygen molecule is destined to receive electrons from the end of the mitochondrial chain, completing the process that began with the first step of glycolysis. The air you inhale becomes the final participant in the story of energy, and the air you exhale carries away the carbon dioxide released in this transformation. In the quiet depths of your cells, these reactions unfold continuously. Pyruvate crosses into the mitochondrion, electrons flow, protons move, and ATP is born anew. The process is never rushed, never chaotic. It moves with the slow precision of something ancient and inevitable. Through it, life renews itself, breath by breath, molecule by molecule. The transition to the mitochondrion is therefore not only a biochemical step, but a reflection of life's deeper rhythm, a movement inward, toward the core of existence, where the invisible work of energy sustains the visible world. 8. Deep inside the mitochondrion, past its folded membranes, and into the calm, enzyme-rich matrix, lies a process of quiet precision, a circular sequence of reactions that fuels all living things. This is the citric acid cycle, known more poetically as the Krebs cycle, after the scientist who first described its pattern. It is a circle of transformation, where the remnants of food molecules are broken down completely, their energy released in careful steps, and their carbon atoms set free to return to the atmosphere. It is not a cycle of destruction but of renewal, one that sustains life through the endless rhythm of conversion, creation, and return. The Krebs cycle begins where glycolysis and the transition step have left off with acetyl-CoA, the two-carbon molecule that represents the distilled essence of glucose. This small molecule enters the matrix like a traveler stepping into a grand system of pathways, ready to surrender its energy to the machinery of life. The first step is adjoining. Acetyl-CoA merges with a four-carbon compound called oxaloacetate, forming a six-carbon molecule known as citrate. It is from this molecule that the citric acid cycle takes its name. This moment marks the beginning of the cycle's graceful rotation, a series of chemical transformations that will carry the carbon atoms of acetyl-CoA through a journey of oxidation, energy capture, and release. Citrate is gradually rearranged and reshaped by enzymes, each reaction preparing the molecule for the next. Along the way, two carbon atoms are removed as carbon dioxide, the same gas that will soon leave the body through exhalation. In losing these atoms, the molecule releases high-energy electrons, which are captured by carrier molecules, NAD mice, and FAD, to form NADH and FADH2. These carriers hold the true energy of the cycle, storing it temporarily until it can be used to generate ATP in the final stages of respiration. At first glance, the Krebs cycle might seem like a mere sequence of reactions, a mechanical loop repeating endlessly within each cell. But on closer reflection, it becomes clear that it is something much more profound, a molecular engine of extraordinary elegance. The oxaloacetate molecule that starts the process is regenerated by the end, ready to join with a new acetyl-CoA and begin again. This self-sustaining rhythm ensures that as long as nutrients and oxygen are available, the cycle never stops. It spins tirelessly, one rotation after another, producing the energy and materials necessary for life. Each round of the Krebs cycle yields important results. 3 molecules of NADH, 1 of FADH2, 1 of ATP, or its close equivalent GTP, and 2 molecules of carbon dioxide. When considered in isolation, these numbers may seem small, but when multiplied by the trillions of mitochondria operating in a human body, the magnitude becomes almost unfathomable. Every heartbeat, every breath, every thought is powered by the collective turning of countless Krebs cycles within your cells, each one running in perfect silence. The beauty of this system lies in its symmetry and its connection to all other metabolic processes. The Krebs cycle is not only a means of breaking down molecules for energy, but also a hub for building new ones. The intermediates formed along its path can be diverted to synthesize amino acids, lipids, and nucleotides, the building blocks of proteins, membranes, and DNA. Thus, while the cycle's primary role is energy extraction, it also provides the raw materials for growth and repair. It stands at the crossroads of metabolism, linking the pathways that break molecules down with those that build them up again. One of the quiet marvels of the cycle is its efficiency. Every atom of the acetyl group, from acetyl-CoA, is accounted for, every electron captured and carried away for use elsewhere. The enzymes that guide each reaction are precise and tireless, passing substrates and products like dancers exchanging hands. There is no waste, no randomness. The entire process is a study in biochemical balance, refined through billions of years of evolution. Even small disturbances, a missing enzyme, a blocked reaction, can throw the cycle into disarray, leading to fatigue, illness, or worse. This fragility serves as a reminder of the delicate harmony upon which life depends. The carbon dioxide produced by the Krebs cycle may seem insignificant, a mere byproduct of respiration, yet it plays a vital role in Earth's grander cycles. The carbon atoms released from your cells once belonged to the food you ate, grains, fruits, or perhaps the flesh of another animal. Before that, they were part of plants, drawn from the atmosphere during photosynthesis. When you exhale, you return them to that same air, where they will once again be absorbed by leaves, transformed by sunlight into sugar, and re-enter the cycle of life. In this way, every breath connects you to the living world, a quiet exchange between your body and the earth, mediated by the ancient chemistry of the Krebs cycle. In the mitochondrion, the NADH and FADH, two molecules produced by the Krebs cycle, do not linger long. They are drawn toward the inner membrane, where they will donate their high-energy electrons to the electron transport chain. There, their captured energy will drive the production of vast quantities of ATP, the ultimate purpose of respiration. The cycle itself, however, remains in constant motion, feeding these carriers into the system as fast as they can be regenerated. Like a wheel turning an unseen generator, the Krebs cycle's steady spin provides the momentum for the next great phase of energy creation. If one could shrink small enough to witness the cycle in motion, it would not appear as a series of static steps, but as a fluid choreography. Molecules would drift, collide, and transform in gentle succession, guided by enzymes that seem almost alive in their precision. The matrix would shimmer with activity, hydrogen ions shifting, carbon dioxide forming and departing, energy moving invisibly from one form to another. There would be no noise, no light, only the quiet, rhythmic pulse of reactions unfolding in timeless sequence. This process is so fundamental, so essential, that it occurs in nearly all aerobic organisms. From the smallest algae floating in the sea to the most complex mammals, the same cycle spins within their cells. Even in organisms that have evolved unusual metabolic pathways, those that live without oxygen or those that use sulfur or iron instead, the traces of the Krebs cycle can still be found, modified but recognizable. It is as though life, once it discovered this pattern, never let it go. There is a kind of poetry in this circularity. The Krebs cycle reflects the very pattern of nature itself, the rhythm of birth, growth, decay, and renewal that governs all living systems. Just as the seasons turn, so too does this molecular wheel, endlessly returning to its beginning, even as it sustains the ongoing flow of life. The carbon atoms that pass through it are like travelers moving through a vast system of exchange, never lost, only transformed. Within you, at this very moment, countless Krebs cycles are turning in silence. They do not wait for your attention, nor do they depend on your will. They continue whether you wake or sleep, maintaining the energy that keeps your heart beating and your mind aware. Every rotation of these cycles releases electrons that will soon power the creation of ATP, ensuring that your body's quiet fire never goes out. The Krebs cycle is not dramatic, nor is it loud. Its work is steady, deliberate, and profoundly necessary. Through it, energy flows seamlessly from the food you eat into the movements and thoughts that define your life. It is a testament to the subtle elegance of biology, a reminder that even in the smallest, most hidden processes, there is an order and grace that sustains everything. In the stillness of life's chemistry, this circular engine continues to turn, never tiring, never stopping. It transforms the remnants of what was once sunlight and matter into the energy of motion and warmth, linking you to the world around you through every breath and every heartbeat. In the unending quiet of your cells, the Krebs cycle keeps its rhythm eternal, patient, and full of life. 9. Electron transport and oxidative phosphorylation. Deep within the folds of the mitochondrion, beyond the quiet turning of the Krebs cycle, lies the final and most magnificent act of cellular respiration, the stage where energy is transformed into its most usable form. Here, on the inner membrane, a delicate chain of proteins works in seamless harmony to create the majority of the cell's ATP. It is a process known as oxidative phosphorylation, powered by the electron transport chain, and it represents one of nature's most intricate and elegant achievements. Through this invisible mechanism, The energy once held in food is refined into the steady current that sustains every heartbeat, every breath, and every moment of awareness. The story of oxidative phosphorylation begins with the electron carriers produced earlier in the process of respiration. From glycolysis, the link reaction, and the Krebs cycle come the molecules NADH and FADH, two small, powerful shuttles loaded with high-energy electrons. These electrons are the true currency of energy, gathered patiently from the breakdown of glucose. Their journey now leads them to the inner mitochondrial membrane, where a series of protein complexes awaits to receive them. These complexes, numbered I through IV, are embedded like shining sentinels along the membrane's surface, each one passing electrons to the next in a carefully controlled sequence. When NADH arrives at complex I, it releases its electrons, returning to its oxidized form, NAD-, ready to be used again. The electrons enter the complex and begin their descent through a series of redox reactions, moving to slightly lower energy states at each step. The energy released by their movement is not wasted. It is used to pump protons, hydrogen ions from the mitochondrial matrix into the intermembrane space. This pumping action creates a gradient, a subtle imbalance that stores potential energy, much like water accumulating behind a dam. FADH2 joins the process at complex 2, donating its electrons slightly further down the chain. These electrons also travel through the membrane complexes, contributing to the same growing reservoir of potential energy. As the electrons pass from one complex to another, they move through small molecular intermediates such as coenzyme Q and cytochrome C. These carriers glide between complexes, ensuring the smooth transfer of electrons while maintaining the chain's rhythm. The electron's path is not random, but exquisitely organized, with each step fine-tuned to release just the right amount of energy. The final destination for these electrons lies at complex 4, where they meet the molecule that gives oxidative phosphorylation its name, oxygen. Here, oxygen acts as the final electron acceptor, combining with electrons and protons to form water. It is a quiet, almost invisible reaction. Yet without it, the entire chain would come to a standstill. This is why oxygen, so easily taken for granted, is the key that keeps the fire of life burning. The gradient created by the pumping of protons across the inner membrane is known as the proton motive force. It represents stored potential energy, an electrochemical tension waiting to be released. The concentration of protons in the intermembrane space becomes higher than in the matrix, and because protons carry a positive charge, an electrical difference also forms. The mitochondrion now holds both a chemical and electrical gradient, a delicate balance of forces that yearns to equalize. The only way for protons to return to the matrix is through a remarkable enzyme called ATP synthase, a molecular machine that harnesses their movement to produce ATP. ATP synthase is one of the most extraordinary structures in biology. It spans the inner mitochondrial membrane, forming a tiny rotary motor powered by the flow of protons. As protons pass through the enzyme, they cause part of it to spin an elegant rotation that drives the chemical joining of ADP and inorganic phosphate to create ATP. Each full turn of this molecular turbine generates 3 molecules of ATP, and within each mitochondrion, thousands of these enzymes work simultaneously, turning ceaselessly as long as the proton gradient persists. The process is breathtakingly efficient, producing around 30 molecules of ATP from the oxidation of a single molecule of glucose. This stage of respiration, though complex, is governed by simple physical principles. Energy flows from high to low, electrons fall through the chain. The ATP produced here powers every form of biological work: the contraction of muscles, the transmission of nerve signals, the repair of DNA, the synthesis of proteins, and the maintenance of ion balances across membranes. It fuels both motion and thought, giving life its continuity and its quiet persistence. The entire system functions with a kind of silent perfection. Each component knows its place, each electron its path, each proton its moment to move. Even the heat released during this process serves a purpose, helping to maintain the body's warmth. The gentle temperature that radiates from living tissue is the whisper of oxidative phosphorylation, the warmth of electrons falling through the chain and of protons returning home through ATP synthase. The process never rests, never falters. It flows like an unbroken current beneath the surface of life. When oxygen supply is interrupted, however, the chain can no longer continue. Without oxygen to receive electrons at the end, the entire sequence backs up like a halted river. NADH and FADH2 cannot unload their electrons, and the flow of protons ceases. ATP production slows, and the cell must rely again on glycolysis, a far less efficient means of generating energy. This shift, though temporary, reveals how dependent modern life is on oxygen, the invisible partner of every living cell. The breath you take in at this very moment sends oxygen molecules on a journey through your bloodstream, across cell membranes, where they complete their purpose with a single act of acceptance, the capture of electrons and the creation of water. From an evolutionary perspective, oxidative phosphorylation represents one of the great turning points in life's history. When oxygen first began to accumulate in Earth's atmosphere billions of years ago, it posed a challenge to early life forms. Oxygen is reactive, even destructive, and many primitive organisms found it toxic. But some cells discovered a way to use this potent molecule safely. Those that mastered this art became the ancestors of modern eukaryotic life, their mitochondria evolving to carry out this oxygen-based respiration with unmatched precision. The world changed forever, and with it, the possibilities for complex, energy-demanding life expanded dramatically. Within every human cell, this ancient process continues unchanged. The food you eat, reduced to its simplest molecules, feeds electrons into the chain. The air you breathe supplies the oxygen to complete the cycle, and the result is life quiet, continuous, and self-sustaining. Each heartbeat, each impulse of thought, each gentle rise of the chest in sleep is powered by this invisible tide of energy flowing through your mitochondria. If one were to imagine this process visually, it would not appear violent or chaotic. It would resemble a slow, glowing current, a cascade of light flowing down the inner membrane. Electrons would drift like fireflies, releasing energy as they pass from one protein to the next. Protons would stream softly through the enzyme, and at the end of it all, a molecule of water would form pure, simple, and silent. This is the true essence of life's energy, a balance between motion and calm, between tension and release, between the unseen and the deeply felt. Oxidative phosphorylation is not just a biochemical event, it is a reflection of life's harmony with its environment. The oxygen drawn from the air and the carbon dioxide released in return bind all living things into one vast cycle, plants giving oxygen, animals returning carbon, the two intertwined in an endless exchange. The mitochondria within you are the continuation of this cosmic rhythm, the meeting point between sunlight captured by plants and the life it sustains in your body. As you sit quietly, perhaps breathing a little more slowly, this process unfolds in every cell without sound or interruption. Electrons glide, protons drift, ATP is formed. It is happening now, in you, and in every living being that shares the air. The electron transport chain hums with the calm assurance of something ancient and eternal, converting the energy of the universe into the warmth of life. And though unseen, its rhythm is steady, the rhythm of existence itself, transforming what was once light and matter into the gentle glow of being. 10. The role of oxygen, the final electron acceptor. Every breath you take carries with it an invisible gift, one that sustains every heartbeat, every spark of thought, and every quiet moment of life. That gift is oxygen, a colorless, tasteless gas that drifts through the air, unnoticed and uncelebrated, yet absolutely essential to existence. It enters your lungs without effort, diffuses into your blood, and travels silently to every corner of your body. There, In the depths of each cell, oxygen performs its most sacred task, accepting the final electrons from the chain of life's energy, completing the process that powers all living beings. Without it, the flow would stop, the chain would break, and the soft hum of cellular activity would fade into stillness. The story of oxygen's role begins deep within the mitochondrion, where the electron transport chain releases energy in small, measured steps. As electrons are passed from one protein complex to another, they gradually lose energy that is used to pump protons across the inner membrane, creating the gradient that drives ATP synthesis. But these electrons, though now lower in energy, cannot simply disappear. They must go somewhere, must find a final resting place where they can come to equilibrium. This is where oxygen steps in, serving as the ultimate electron acceptor, the quiet receiver at the end of the chain, who ensures that the flow continues without interruption. When the electrons reach the final complex of the chain, they are transferred to molecules of oxygen, which combine with hydrogen ions to form water. This single, unassuming reaction, the union of electrons, protons, and oxygen, is what allows life's energy systems to continue cycling. It is both the end and the beginning of the process, completing one round of respiration while setting the stage for another. The water formed through this reaction is not waste. It is a byproduct of creation itself, the gentle signature of balance restored. In every drop of water produced by your mitochondria lies the story of oxygen's participation in the flow of life. The need for oxygen in this process reveals something profound about living systems. Oxygen does not create energy. It allows energy to be released in a controlled and sustainable way. Without it, electrons would have no final destination. The electron transport chain would stall, and ATP production would cease. The cell, deprived of its power source, would quickly run out of energy and falter. This is why, when oxygen supply is cut off, even for a few minutes, the brain, which relies on a constant supply of ATP to maintain its delicate electrical activity, is especially sensitive. It can tolerate only the briefest interruption before its functions begin to deteriorate. Oxygen, therefore, is not a luxury. It is the quiet thread that holds consciousness and motion together. To understand why oxygen is so effective at this role, one must look at its nature. Oxygen is a highly electronegative element, meaning it has a strong tendency to attract electrons. This property allows it to pull electrons through the entire chain, ensuring that the flow never reverses or stalls. In essence, oxygen acts like a magnet at the end of a long corridor, drawing electrons forward until they finally settle into its embrace. The beauty of this arrangement lies in its balance. Oxygen's strength is just right powerful enough to attract the electrons, but not so strong as to cause chaos within the system. It creates order from potential, guiding the invisible current of energy to its final, peaceful conclusion. The arrival of oxygen on early Earth changed everything. For the first billion years or so, the planet's atmosphere contained almost no free oxygen. Life during that time was simple, limited to anaerobic organisms that relied on less efficient forms of energy extraction, such as fermentation. These early creatures thrived in a world without air, but their energy yield was small and their possibilities limited. Then, through the evolution of photosynthesis, some organisms began to release oxygen as a byproduct. Over millions of years, this gas accumulated in the atmosphere, transforming the planet's chemistry and paving the way for new forms of life. What was once a toxic byproduct to many early microbes became the foundation of complexity itself. When life learned to use oxygen as the final electron acceptor, the energy yield of respiration increased dramatically. The oxidation of a single molecule of glucose in the presence of oxygen can produce around 30 molecules of ATP, nearly 15 times more than anaerobic processes. This abundance of energy allowed cells to grow larger, specialize, and combine into multicellular organisms. Muscles, nerves, and organs All the hallmarks of complex life owe their existence to the ability to harness oxygen safely. It was a turning point not just in biology, but in the destiny of the planet itself. Yet oxygen is both a giver and a taker. Its reactivity, the very property that makes it so valuable in energy metabolism, also makes it potentially dangerous. As electrons move through the transport chain, a small fraction may escape prematurely, reacting with oxygen to form reactive oxygen species, unstable molecules that can damage DNA, proteins, and membranes. To counter this, cells have evolved sophisticated defense systems, including enzymes like superoxide dismutase and catalase, which neutralize these harmful byproducts. The balance between oxygen's creative and destructive potential is one of the great achievements of evolution, a delicate truce that allows life to flourish without self-destruction. The dependence on oxygen connects all living things in an unbroken chain of exchange. Plants, through photosynthesis, take in carbon dioxide and release oxygen. Animals, in turn, take in that oxygen and release carbon dioxide. Each breath you exhale becomes part of this global rhythm, returning carbon to the atmosphere where it can once again be absorbed by the green world. The oxygen you breathe may have been released by a leaf only hours ago, or by a tree that stood a continent away. It may even come from the ocean, where plankton, invisible to the naked eye, produce more than half of the world's oxygen. Every inhalation, therefore, is a quiet communion with the rest of life, a participation in a cycle that spans the earth itself. Inside your body, this same pattern repeats on a smaller scale. Oxygen enters the lungs, binds to hemoglobin in the blood, and is carried through the arteries to every organ. It moves from one environment to another, from the vast openness of the atmosphere to the narrowest capillaries of the body. Finally, it diffuses into the cells, slips across the mitochondrial membrane, and finds its place at the end of the electron transport chain. There, in the quiet of the matrix, it performs its timeless duty, accepting electrons, forming water, and allowing the cycle to continue. It is a journey both physical and spiritual, a reminder of how intimately the act of breathing is tied to the essence of life. When you take a slow breath, perhaps without even noticing, oxygen flows into your body, carried by air that has been circulating the planet for millions of years. Each molecule of oxygen you inhale may have once passed through the lungs of another creature, or drifted over oceans, forests, and mountains. It is part of a shared atmosphere that connects every living being that has ever lived. The oxygen that now sustains your cells is the same element that once fed the mitochondria of your ancestors, the same force that fuels every living creature today. In this way, breathing is not just a personal act, it is a participation in the long continuity of life itself. If one listens carefully to this idea, it becomes almost poetic. Every exhalation is an offering, every inhalation a gift received in return. The oxygen you breathe becomes part of you for a brief moment, helping to create the ATP that powers your body and mind, and then it leaves again, transformed into carbon dioxide and water, ready to rejoin the great circulation of nature. The molecules of water produced by your mitochondria may one day rise as vapor, fall again as rain, and nourish the very plants that release the oxygen you will breathe tomorrow. There is no separation, only flow, only exchange. In the stillness of night, as your body rests and your breathing becomes slow and even, this process continues without thought or effort. Oxygen enters, electrons fall, and water forms. It happens in silence, unseen yet unceasing. The mitochondria within your cells, like tiny flickering lanterns, continue their quiet labor, using oxygen to keep the flame of life alight. Each molecule of oxygen fulfills its purpose perfectly, receiving what was once energy from the sun and returning it gently to the world. Oxygen is not simply a gas. It is the quiet ending of one journey and the beginning of another. It is the final receiver in the chain of life's energy, the element that completes the circle and allows existence to continue unbroken. Every breath you take affirms this truth that life endures because of balance, because of the calm acceptance of what must pass from one form to the next. And as long as oxygen flows, so too does life, steady and serene, carried forward by the endless rhythm of respiration that binds all living things together. 11. Anaerobic pathways, life without oxygen. Long before oxygen filled the skies, before forests spread across continents and animals walked upon the land, life was already thriving in the quiet darkness of the ancient seas. The atmosphere then was heavy with carbon dioxide and methane, and the air as we know it, rich in oxygen and fit for breathing, did not yet exist. Yet, in that oxygenless world, the earliest organisms discovered ways to live and grow. They relied on the chemistry of Earth itself, drawing energy not from oxygen but from other reactions that could yield just enough power to sustain existence. These were the first anaerobes creatures that lived without air, and even now, billions of years later, their descendants remain among us, quietly demonstrating that life can persist even in places where oxygen cannot reach. In the modern cell, oxygen-dependent respiration is the most efficient way to produce energy, generating large amounts of ATP through the combined efforts of the Krebs cycle and oxidative phosphorylation. But when oxygen becomes scarce or absent, cells can turn to their older, simpler methods. They provide a temporary or alternative means of generating ATP when oxygen is unavailable, ensuring that the essential functions of life can continue, at least for a while. Anaerobic energy production begins in the same place as aerobic respiration with glycolysis. This pathway, taking place in the cytoplasm, splits glucose into two molecules of pyruvate while producing a small but vital supply of ATP. The process also creates NADH, a molecule that carries electrons and must be recycled to NAD mice for glycolysis to continue. Under normal aerobic conditions, NADH donates its electrons to the electron transport chain in the mitochondrion, where oxygen accepts them and energy is extracted efficiently. But when oxygen is absent, the chain halts and NADH accumulates. Without a way to regenerate NAD mice, glycolysis would stop, and the cell would quickly exhaust its energy supply. To prevent this, cells use fermentation, a simpler, oxygen-independent process, to restore the balance and keep ATP production flowing. In human and animal cells, this occurs through lactic acid fermentation. Here, the pyruvate produced during glycolysis accepts electrons from NADH, converting back into NAD mice and forming lactic acid as a byproduct. This reaction allows glycolysis to continue cycling, producing ATP even without oxygen's help. The yield is small, only 2 ATP molecules per glucose, but it can sustain cells for short periods of intense activity or limited oxygen availability. In muscle tissue, this process becomes especially important during bursts of exertion, when energy demand outpaces oxygen supply. The burning sensation that sometimes accompanies strenuous exercise is a result of lactic acid accumulation, a reminder that even within the human body, the ancient anaerobic pathways still awaken when needed. Once oxygen becomes available again, the lactic acid is gradually converted back to pyruvate, which then enters the mitochondria to complete its oxidation through aerobic respiration. The temporary debt incurred during anaerobic metabolism is repaid as the body returns to balance. This return to normal breathing, the deep intake of air after exertion, is not just recovery, it is the restoration of the cell's most efficient energy system. The cycle of oxygen and its absence of anaerobic and aerobic processes reflects the adaptability of life, the seamless shifting between old and new mechanisms of survival. In microorganisms, however, anaerobic life is not merely a backup plan. It is a way of being, Certain bacteria and archaea thrive in environments devoid of oxygen deep in oceanic sediments, inside volcanic vents, within animal intestines, and in the quiet mud at the bottom of lakes. These organisms use alternative electron acceptors in place of oxygen, such as nitrate, sulfate, or even carbon dioxide. The products of their metabolism vary widely: methane, hydrogen sulfide, and other gases that give Earth's depths their distinctive odors. Though their chemistry differs, their purpose is the same: to maintain the flow of electrons, to capture energy, to keep the pulse of life moving, even in the most inhospitable conditions. Among the most fascinating of these are methanogens, ancient microorganisms that produce methane as a byproduct of their metabolism. They exist today in environments as extreme as deep-sea hydrothermal vents and as familiar as the digestive systems of cattle. Their presence connects the distant geological past to the present day, reminding us that the chemistry of life is vast and varied. In another branch of anaerobic life, sulfate-reducing bacteria carry out reactions that produce hydrogen sulfide. These organisms play crucial roles in recycling nutrients in ecosystems, breaking down organic matter and returning elements to the environment for reuse. Though they live far from the sunlight and oxygen that sustain most of life, they are no less integral to the balance of the biosphere. Anaerobic pathways are also responsible for many of the transformations humans have learned to harness for their own use. Yeast, for example, perform alcoholic fermentation, a process that converts pyruvate into ethanol and carbon dioxide. This ancient chemistry has been part of human culture for thousands of years, giving rise to bread, beer, and wine. When yeast cells run out of oxygen, they shift seamlessly to fermentation, releasing bubbles of carbon dioxide that make dough rise and producing ethanol that enriches fermented beverages. In every loaf of bread and every glass of wine lies the same fundamental process that sustains life in oxygen-free environments, the timeless art of extracting energy from sugar without the aid of air. The difference between aerobic and anaerobic metabolism is, in many ways, aerobic respiration, with its reliance on oxygen, is like a steady, bright flame, capable of producing large amounts of energy over long periods. Anaerobic pathways, by contrast, are like a brief spark, limited but immediate, a rapid response to sudden need or environmental constraint. In human muscle, both systems work together in harmony. The anaerobic system provides the first burst of energy for movement, while the aerobic system gradually takes over as oxygen delivery catches up. This partnership between the ancient and the modern, the simple and the complex, is what allows life to adapt so seamlessly to changing conditions. From an evolutionary perspective, the persistence of anaerobic pathways reveals the deep continuity of life's chemistry. Though the Earth's atmosphere has long been rich in oxygen, cells have never abandoned their older mechanisms. Instead, they have layered new processes on top of old ones, preserving each adaptation that once proved useful. This layering of history within biology, the coexistence of the ancient and the advanced, gives life its extraordinary flexibility. A cell today carries within it the echoes of its ancestors, the enzymes of glycolysis, the reactions of fermentation, and the intricate systems of oxygen-based respiration, all coexisting within a single organism. Anaerobic metabolism also reminds us that life is not confined to a narrow set of conditions. Wherever energy can be captured, life finds a way. From the frozen lakes of Antarctica to the scalding springs of volcanic craters, from the deepest caves to the interiors of animal bodies, anaerobic organisms persist. They teach us that oxygen, while central to our own survival, is not the only path to vitality. In the grand scheme of the biosphere, anaerobic processes continue to power a hidden world, a vast, unseen network of microbial life that recycles carbon, nitrogen, and sulfur, sustaining ecosystems from below. When we breathe, we live in partnership with both sides of this story. The oxygen we inhale supports our mitochondria, while the carbon dioxide we exhale feeds the plants and microbes that may, in turn, live without oxygen. The cycle is whole, inclusive, and eternal. In this exchange, the anaerobic and the aerobic are not opposites, but complements, two halves of the same living balance. As you rest, your own cells continue this dance between oxygen-rich and oxygen-poor metabolism. In moments of calm, oxygen flows freely, and your mitochondria hum with quiet efficiency. But in fleeting instants, the blink of an eye, the beat of a muscle, small pockets within you return to the ancient way, drawing briefly on fermentation to bridge the gap. You carry within your very tissues the memory of a world without air, an echo of the first living cells that learned to survive in darkness. To reflect on this is to glimpse the profound continuity of life. Every breath, every movement, every moment of stillness is sustained by chemistry that has persisted for billions of years. Oxygen may be the air of life, but the ability to live without it lies deep within our biological heritage. It is a quiet reminder that life does not depend on one condition alone, but on the endless adaptability of its own design. In this adaptability lies resilience, the capacity to endure, to persist, and to continue the gentle, unbroken rhythm of existence, with or without the air we breathe. 12. In the quiet workings of the cell, energy is never wasted, and no potential source is left unused. While glucose serves as the body's primary and most immediate fuel, life's machinery is versatile and wise. It knows how to draw from other reserves, from the stored richness of fats and the steady structure of proteins when circumstances require. These molecules, though not always the first to be used, are vital to the continuity of life. They form the deeper wells of energy that sustain the body through fasting, exertion, or scarcity. Within them lies a slower, more enduring form of power, a quiet strength drawn upon when the simpler sugars have already been spent. When you think of energy, it is easy to imagine the sweetness of glucose, the immediacy of its combustion within the cell. Yet fats, though less conspicuous, hold far more energy in their bonds. A single gram of fat provides more than twice the energy of a gram of carbohydrate or protein. This efficiency is no accident. It is a result of the very nature of fat's chemistry. The long chains of carbon and hydrogen that make up fatty acids are rich in reduced bonds, each one capable of releasing electrons when broken down. These electrons, in turn, feed into the same pathways that process glucose, ultimately producing ATP through the familiar machinery of the mitochondrion. The story of fat as fuel begins with the process of lipolysis, the gentle breakdown of triglycerides, which are the body's main form of stored fat. Each triglyceride consists of 3 fatty acids attached to a molecule of glycerol. When energy demand rises, enzymes within fat cells cleave these bonds, releasing the fatty acids into the bloodstream. Carried by proteins, these molecules travel to tissues in need muscles, the heart, or the liver, where they are taken up and prepared for oxidation. The glycerol portion, though smaller, can also enter the energy pathways, being converted into an intermediate of glycolysis and continuing toward ATP production. Inside the cell, the released fatty acids must first undergo activation, a process that attaches them to coenzyme A, forming fatty acyl-CoA. This activated molecule is then transported into the mitochondrion through a specialized system involving carnitine, a small but essential molecule that acts as a shuttle, ensuring that fatty acids cross the inner mitochondrial membrane. Once inside, the real transformation begins in a process known as beta oxidation. Beta oxidation is a rhythmic sequence of reactions that systematically shortens the fatty acid chain, removing 2 carbon atoms at a time. Each turn of this process produces 1 molecule of acetyl-CoA, which then enters the Krebs cycle, just as it would from the breakdown of glucose. In addition, each round generates 1 molecule of NADH and 1 of FADH2, both of which feed electrons into the electron transport chain to produce more ATP. The beauty of this system lies in its continuity. Though fats are structurally different from sugars, the cell processes them through the same central pathways, using the same molecular engines. Energy, regardless of its source, always finds its way to ATP. The yield of energy from fat oxidation is immense. A single molecule of palmitic acid, a common fatty acid found in the body, can produce more than 100 ATP molecules. When fully oxidized, far more than the 30 or so ATP molecules generated from a molecule of glucose. This is why fat serves as the body's long-term energy reserve, stored quietly in adipose tissue to be drawn upon when needed. It is dense, efficient, and stable, capable of sustaining life during long periods without food. When you fast, exercise intensely, or experience cold, your cells gently begin to turn to these reserves, breaking down fat to maintain warmth and motion. The metabolism of fat is not uniform across the body. Different tissues rely on it to varying degrees. The heart, for instance, is especially dependent on fat as a fuel source, deriving most of its energy from fatty acid oxidation, even under normal conditions. Muscles, too, draw upon fat during prolonged, moderate activity, conserving glucose for moments of higher intensity. The liver plays a central coordinating role, converting fatty acids not only into ATP but also into ketone bodies, small, energy-rich molecules that can be sent into the bloodstream to nourish other tissues, including the brain, when glucose is scarce. These ketone bodies, produced during fasting or carbohydrate restriction, serve as a gentle alternative to glucose, ensuring that even in times of shortage, the energy supply remains unbroken. Yet, while fat provides endurance and abundance, protein stands as the reserve of last resort. Proteins are the building blocks of the body, the fabric of muscles, enzymes, and structures. They are not meant to serve as fuel, and yet, in times of prolonged deprivation or extreme exertion, the body can turn to them when other sources have waned. This transformation is known as protein catabolism, and it is a process both efficient and solemn, as the body begins to recycle its own materials to preserve life. In protein metabolism, enzymes break down complex proteins into their individual amino acids. Some of these amino acids can be converted into intermediates that feed into glycolysis or the Krebs cycle, depending on their structure. Amino acids, whose carbon skeletons can become pyruvate or oxaloacetate, are called glucogenic, as they can contribute to glucose formation through gluconeogenesis, the creation of new glucose molecules in the liver. Others, called ketogenic amino acids, are converted into acetyl-CoA or acetoacetate, joining the pathways of fat metabolism. In this way, proteins, too, can ultimately provide energy, though at the cost of cellular material. The nitrogen atoms of amino acids, which cannot be oxidized for energy, are removed in the process of deamination. This produces ammonia, a toxic compound that the body quickly converts into urea in the liver. The urea is then excreted safely through the kidneys. Even here, the body's precision is evident. Nothing is wasted, and every step is managed with balance and care. The breakdown of proteins for energy is a last measure, a system designed to ensure survival when all else fails. It is not ideal, but it is life's way of protecting itself under duress. Together, the oxidation of fats and the metabolism of proteins complete the picture of cellular energy. They remind us that life's chemistry is profoundly interconnected, that no molecule exists in isolation, and that the pathways of energy weave through every corner of metabolism like threads in a tapestry. When glucose runs low, fats take its place. When fats diminish, proteins offer their structure. The flow continues, adapting to every change, maintaining the equilibrium that defines life. From an evolutionary perspective, this flexibility is one of the great triumphs of biology. Early life forms that could use multiple energy sources had an advantage in a world where conditions changed constantly. That adaptability has been preserved in every organism since. In the cells of your body, the same capacity remains an inheritance from ancestors who survived in scarcity and abundance alike. This constant interchange between sugars, fats, and proteins is more than a chemical necessity. It is a reflection of life's balance. In times of plenty, energy is stored as fat, patiently waiting for a future need. In times of need, those reserves are gently released, transformed back into the power that moves muscles and warms blood. Even as you rest, this quiet exchange continues, molecules flowing, bonds breaking and reforming in endless succession. In the stillness of your body, this process is invisible, yet ever present. The fats stored beneath your skin and within your cells, the proteins within your muscles and organs, all are part of a living reservoir of energy, ready to serve when called upon. The mitochondria, ever faithful, continue their work, transforming these molecules into ATP, the quiet currency of life. Nothing is hurried, nothing is wasted. The same carbon atoms that once formed part of a meal, or a plant, or an animal, are now part of you, and in time, they will return to the world again, to nourish other lives in the same endless cycle. In this continuity, there is a quiet beauty. The body, like the earth itself, depends on cycles of renewal and exchange. Fats, proteins, and sugars flow through you as sunlight made tangible, each one a different expression of the same fundamental energy. You are not separate from this flow, you are part of it, sustained by it, and, in turn, contributing to it with every breath, every movement, every quiet moment of being. The chemistry that keeps you alive is not only efficient and elegant, it is deeply harmonious, a reminder that life itself is the art of transformation. 13. The mitochondrion in detail, structure, and purpose. Within the quiet architecture of every living cell, there exists a realm devoted entirely to the transformation of energy, a place where the raw materials of life are refined into the invisible current that sustains motion, thought, and renewal. This realm is the mitochondrion, the organelle often called the powerhouse of the cell. Yet, this description, while true, only hints at its deeper significance. The mitochondrion is not merely an engine, but a living relic of Earth's ancient past, a self-contained system of exquisite design. It is both a factory of energy and a guardian of balance, orchestrating the rhythms of respiration that give rise to life's steady pulse. Each mitochondrion is a world unto itself, a small, double-membrane structure suspended in the cytoplasm like a lantern glowing softly in the dark. Some cells contain only a few, while others, especially those that demand constant energy, such as muscle and nerve cells, contain thousands. Their number and activity reflect the life of the cell they serve. The more energy required, the more mitochondria present. Yet no matter how many dwell within a cell, each one carries out the same essential task: the continual conversion of nutrients into ATP, the energy currency of life. To understand how the mitochondrion performs this role, one must first look closely at its structure. It is enclosed by two membranes, each with its own properties and purpose. The outer membrane is smooth and porous, allowing small molecules to pass through freely. It acts as a gentle barrier, defining the organelle's boundary while maintaining openness to the surrounding cytoplasm. The inner membrane, by contrast, is highly selective and intricately folded, forming countless inward curves known as cristae. These folds dramatically increase the surface area available for the reactions of respiration, giving the mitochondrion the appearance of a labyrinth of fine ridges under the microscope. Between these two membranes lies the intermembrane space, a narrow compartment that plays a crucial role in the movement of protons during oxidative phosphorylation. Deep within, the innermost region, the matrix serves as the heart of the organelle. It is here that the citric acid cycle unfolds, enzymes catalyzing reactions that generate the high-energy molecules NADH and FADH2. The matrix contains not only these enzymes, but also ribosomes, ions, and a small ring of mitochondrial DNA, a genetic echo of the organelle's ancient independence. This DNA is one of the mitochondrion's most remarkable features. Unlike most cellular components, which rely entirely on the nucleus for their genetic instructions, mitochondria possess their own genome. It is small, circular, and remarkably similar to the DNA found in bacteria. This similarity is no coincidence. It is evidence of the mitochondrion's origin as a free-living organism. Billions of years ago, an ancestral cell engulfed a smaller bacterium capable of using oxygen to extract energy efficiently. Instead of being digested, the bacterium survived within its host, forming a symbiotic relationship. Over immense spans of time, this partnership deepened until they became inseparable, the bacterium evolving into the mitochondrion, the cell evolving into the eukaryote. This event, known as endosymbiosis, marks one of the most profound turning points in the history of life. From it arose the complexity of plants, animals, and all multicellular beings. The mitochondrion, though now dependent on its host cell, retains traces of its ancient autonomy. It divides independently, replicating much like bacteria through a process of fission. Each new cell inherits mitochondria from its parent, and in humans, this inheritance occurs almost entirely through the mother. Within every cell of your body, the mitochondria you possess are direct descendants of those carried by your maternal ancestors, stretching back through time to the first humans and beyond. The purpose of the mitochondrion extends far beyond simple energy production. While it is true that its main task is to synthesize ATP, the processes it governs ripple outward through nearly every aspect of cellular life. The mitochondrion manages calcium levels, a function essential for communication between cells and the contraction of muscles. It participates in the synthesis of certain hormones and amino acids. It also plays a role in determining the fate of the cell itself, regulating programmed cell death, or apoptosis, the orderly process by which old or damaged cells are removed to make way for new ones. In this sense, mitochondria are custodians of both life and death, maintaining balance through their silent governance. At the molecular level, the inner membrane's cristae form the stage on which the electron transport chain and ATP synthase operate. Here, electrons flow along a series of complexes, and protons are pumped across the membrane, creating the electrochemical gradient that drives ATP synthesis. Each crista is like a winding corridor filled with these molecular machines, tirelessly spinning and shuttling electrons in perfect synchrony. The efficiency of this arrangement is breathtaking. The more folds a mitochondrion has, the greater its surface area, and the more ATP it can produce. This is why highly active tissues, such as the heart or the brain, possess mitochondria with densely packed cristae, each one a powerhouse of activity. Despite their endurance, mitochondria are not immutable. They are dynamic, constantly changing shape through processes called fusion and fission. They merge to share contents and repair damage, then divide to create new organelles or to respond to the cell's shifting needs. This balance between joining and separating allows the mitochondrial network to adapt to stress, demand, and age. Even within a single cell, mitochondria are in motion, drifting, elongating, fragmenting, and reforming in a quiet dance that mirrors the living rhythm of the organism itself. Their central role in metabolism also makes mitochondria key players in aging and disease. When the electron transport chain leaks small amounts of reactive oxygen species, the resulting oxidative stress can damage mitochondrial DNA and proteins. Over time, this accumulation of damage may impair energy production, contributing to the gradual decline associated with aging. Many disorders from metabolic syndromes to neurodegenerative diseases have roots in mitochondrial dysfunction. Yet even here, there is balance. The cell's mechanisms of repair and renewal continually replace old mitochondria with new ones, preserving vitality for as long as possible. The interplay between mitochondria and the rest of the cell is one of cooperation and constant dialogue. The nucleus encodes most of the proteins the mitochondrion needs, while the organelle provides the energy that sustains nuclear functions. Signals flow both ways, ensuring harmony between energy production and cellular activity. When the cell grows, divides, or responds to stress, its mitochondria adjust accordingly, altering their number and activity to match the cell's requirements. Beyond their biological roles, mitochondria carry a symbolic weight. They embody the essence of interdependence. They remind us that life, even at its smallest scale, thrives through partnership and exchange. The oxygen you breathe, the food you consume, and the energy that sustains you are all connected through the quiet work of these ancient organelles. Every thought you have, every step you take, and every heartbeat you feel depends on the unseen labor of trillions of mitochondria within you, each one performing its task with unwavering precision. To imagine their activity is to envision a universe within the self, a landscape of shimmering membranes and flowing ions, where energy moves like light through a web of connections. Each mitochondrion hums softly with purpose, its inner structures folding and unfolding like the pages of an ancient book, telling the story of life's endurance through time. And though they are invisible to the naked eye, their collective presence forms the foundation of existence. In the silence of your body, as you rest or breathe, the mitochondria continue their tireless work. They take the carbon from your food, the oxygen from your lungs, and unite them in a process that releases the warmth that keeps you alive. In doing so, they honor a lineage that began in the dawn of life, billions of years ago, when a tiny cell welcomed another into its embrace and discovered, together, the power to endure. The mitochondrion, then, is more than an organelle. It is a bridge between past and present, between independence and unity, between energy and life. Within its walls, the story of evolution is written in every molecule, and through its ceaseless labor, the flame of existence is kept alight. 14. Cellular, energy balance, and regulation. Within every living cell, a delicate harmony is maintained, a constant balance between energy creation and energy use. This balance, known as cellular energy homeostasis, is the invisible rhythm that sustains life. It ensures that every heartbeat, every breath, every microscopic movement of ions across a membrane happens neither too fast nor too slow, neither in excess nor in deficiency. The cell's world is one of measured precision, where every reaction is tuned to need, and every molecule of ATP is accounted for. Without this balance, even the most intricate biochemical machinery would fall into disorder, and the smooth flow of life would falter. The cell's need for energy never ceases. Whether it is a neuron transmitting an impulse, a muscle contracting, or a liver cell synthesizing enzymes, each task draws upon the same reservoir of chemical energy. ATP, the molecule of life's currency, is constantly being spent and renewed. In a single second, millions of ATP molecules are hydrolyzed in each cell, their stored energy released to power countless reactions. Yet the total quantity of ATP within a cell remains almost constant because it is regenerated just as quickly. The rate at which ATP is made and used is precisely regulated, adjusted to match the cell's changing demands. This regulation is accomplished through feedback, a language of molecules that allows the cell to sense its own state and respond accordingly. When ATP levels fall, signaling molecules such as ADP and AMP rise. These serve as quiet messengers, alerting enzymes and organelles that more energy must be produced. Certain enzymes, like phosphofructokinase in glycolysis, are sensitive to these changes and respond instantly, increasing their activity when energy is low and slowing down when ATP is abundant. In this way, the cell maintains equilibrium, ensuring that energy is produced only when and where it is needed. AMP, the The molecule formed when ATP is used heavily and rapidly plays a particularly important role in this regulation. Its presence activates a master enzyme called AMP-activated protein kinase, or AMPK. This enzyme functions as the cell's energy guardian. When AMPK senses that energy reserves are running low, it triggers a series of adjustments promoting catabolic pathways that generate ATP while suppressing anabolic ones that consume it. It encourages the breakdown of fats and glucose for fuel, while temporarily slowing processes such as protein and lipid synthesis. The result is conservation and restoration. The mitochondria, as the chief producers of ATP, are central to this dynamic system. Their activity rises and falls with the cell's needs, modulated by the supply of oxygen and nutrients. When energy demand increases, as in active muscle cells, mitochondria work at full capacity, their inner membranes humming with the flow of electrons. When demand decreases, their activity slows, conserving oxygen and reducing the production of reactive byproducts. This adaptability gives the cell its resilience, its ability to thrive under fluctuating conditions without waste or shortage. But the regulation of energy is not confined to individual cells. Across the entire body, a vast network of signals coordinates energy balance between tissues and organs. Hormones act as messengers, carrying information about energy status throughout the organism. Insulin, secreted by the pancreas in response to rising blood glucose, promotes the storage of energy by encouraging cells to take up glucose and synthesize glycogen and fat. Glucagon, by contrast, is released when glucose levels fall, stimulating the liver to release stored sugar and prompting fat cells to mobilize their reserves. Together, these hormones maintain the balance between storage and use, between feast and fasting. Other hormones join in this symphony. Adrenaline, produced during moments of stress or exertion, acts swiftly to release glucose and fatty acids into the bloodstream, preparing the body for immediate action. Cortisol, a slower-acting hormone, adjusts metabolism during prolonged stress or fasting, ensuring that the brain and vital organs continue to receive energy. Even leptin and ghrelin hormones secreted by fat tissue in the stomach play roles in signaling hunger and fullness, influencing how much energy the body takes in and expends. Every organ participates in this quiet conversation, guided by the same principle of balance that governs each cell. In a healthy organism, these systems operate with remarkable precision, yet the balance is fragile. When disrupted, energy can accumulate in the wrong places or fail to reach where it is needed. Overnutrition, for example, floods the body with excess glucose and fats, overwhelming regulatory systems and leading to metabolic disorders such as insulin resistance and diabetes. On the other hand, prolonged energy deficiency, as in starvation or chronic illness, forces the body to draw upon its own tissues, consuming fat and muscle alike to sustain essential functions. In both extremes, the fundamental harmony of energy regulation is lost, and with it, the stability of life itself. At the cellular level, disturbances in energy balance can lead to subtle yet far-reaching effects. If mitochondria produce more reactive oxygen species than the cell can neutralize, oxidative stress can damage DNA, proteins, and membranes, gradually impairing the cell's vitality. If ATP levels drop too low, energy-dependent processes from active transport to synthesis begin to fail, leading to cellular fatigue or death. To prevent this, cells maintain multiple layers of protection. They can increase the number of mitochondria through a process called biogenesis, enhance antioxidant defenses, or switch temporarily to alternative energy sources like fats or ketone bodies. These adaptive strategies reveal the depth of life's ingenuity, the ability to restore equilibrium through change. The regulation of energy is also intertwined with time itself. Many aspects of metabolism follow circadian rhythms, rising and falling with the day and night. During waking hours, cells are primed for energy production, and use. During rest, they shift toward repair and renewal. Hormones like insulin and cortisol follow these daily patterns, ensuring that food intake, activity, and energy storage remain synchronized with the body's internal clock. When these rhythms are disrupted by irregular sleep, stress, even at the molecular level, the dance of energy mirrors the larger cycles of the natural world. In this vast web of coordination, energy balance is more than a chemical necessity. It is an expression of order. The body is not a machine running blindly, but a living system that senses, adapts, and harmonizes. The flux of ATP, the pulse of hormones, the breath of oxygen, and the movement of nutrients, all are parts of a continuous flow that mirrors the greater rhythms of life and nature. In moments of exertion, energy surges like a rising tide. In rest, it ebbs gently, restoring calm. Through these fluctuations, stability emerges, not as stillness, but as a constant, graceful adjustment. If one could listen to the sound of this process, it would be like hearing the heartbeat of existence itself, steady, responsive, and endlessly self-correcting. Every cell, from the smallest bacterium to the most complex neuron, each balances its own needs with the demands of the whole, creating an equilibrium that sustains both individuality and unity. This is the true meaning of cellular energy regulation, not a rigid control, but a living balance, flexible, intelligent, and deeply connected to every aspect of life. As you rest, your body continues this silent work. The mitochondria within your cells measure oxygen and fuel, adjusting their output to your state of stillness. Hormones circulate quietly, maintaining warmth, balance, and calm. Even in sleep, energy flows not for motion, but for renewal. Cells repair their structures, store glycogen, and prepare for the coming day. In these hours of rest, energy is not expended but refined, reminding us that balance is not only about activity, but also about restoration. In the end, energy balance is not merely a matter of metabolism. It is the foundation of vitality. It is what allows a single cell to thrive, a body to live, and a world of organisms to coexist in harmony. Every heartbeat, every breath, and every gentle pulse of warmth beneath your skin is the outward expression of this inner equilibrium, a reminder that life endures not through constant motion, but through the art of quiet regulation. 15. Energy in motion, muscles, and movement. Every movement of the body, no matter how subtle or grand, draws upon a vast and intricate network of biochemical reactions. When you lift your hand, take a step, or even breathe, countless muscle fibers awaken, each one fueled by the quiet power of ATP. These movements may seem effortless, yet beneath them lies a symphony of cellular coordination, the conversion of chemical energy into motion, the transformation of invisible molecules into tangible strength. It is in this gentle interplay between biology and physics that the story of energy becomes the story of movement itself. Muscles are remarkable tissues, designed with precision to perform work through contraction. They are made up of long, slender cells called muscle fibers, each one a bundle of contractile units known as myofibrils. Within these myofibrils lie two types of filaments, actin and myosin, that slide past one another to shorten the fiber. This sliding action is the foundation of movement, powered entirely by the hydrolysis of ATP. The process is both mechanical and chemical, a continuous cycle of binding, pulling, releasing, and rebinding that allows a muscle to contract and then relax again. The molecule of ATP is the direct fuel for this cycle. When ATP binds to the myosin head, a small, projecting part of the thick filament, it causes the head to detach from the actin filament. The ATP is then hydrolyzed into ADP and inorganic phosphate, releasing energy. This energy cocks the myosin head into a high-energy position, ready to attach to a new site on the actin filament. When it binds again, the phosphate group is released, and the myosin head pivots, pulling the actin filament inward in what is known as the power stroke. Finally, ADP is released, and a new ATP molecule binds, allowing the process to repeat. In this continuous rhythm of attachment and release, the muscle fiber shortens, and the collective action of millions of fibers results in motion. The body stores only a small amount of ATP in each muscle cell, enough to sustain a few seconds of intense contraction. To maintain movement, ATP must be regenerated almost immediately. The cell accomplishes this through several interconnected systems that ensure a steady supply of energy, each one activating as the previous begins to fade. The first of these systems is the phosphagen system, which relies on a molecule called creatine phosphate. When muscle activity begins, creatine phosphate donates a phosphate group to ADP, rapidly regenerating ATP. This reaction happens almost instantly and provides energy for the first few seconds of movement, making it vital for short bursts of power, such as sprinting or lifting a heavy weight. As this immediate reserve diminishes, the muscle shifts to glycolysis, the breakdown of glucose to form ATP without the need for oxygen. This anaerobic pathway provides energy for up to a few minutes of intense activity, producing small amounts of ATP quickly, but also generating lactic acid as a byproduct. Though often associated with fatigue, lactic acid serves an important role. It allows ATP production to continue when oxygen is scarce, and later, it can be reconverted into pyruvate or glucose once oxygen becomes available again. During sustained activity, when the heart and lungs deliver sufficient oxygen to the muscles, aerobic respiration takes over, using glucose and fatty acids to produce ATP through the mitochondria. This system, though slower, is far more efficient, capable of supporting hours of continuous effort. Muscle fibers themselves are adapted to these different modes of energy production. Some are built for quick, powerful contractions. These are the fast-twitch fibers, rich in enzymes for glycolysis and capable of explosive movement but prone to fatigue. Others, the slow-twitch fibers, are designed for endurance. They contain abundant mitochondria and myoglobin, a pigment that stores oxygen within the cell, giving these fibers their deep red color. Slow-twitch fibers rely on aerobic respiration, burning fuel slowly and steadily, allowing for sustained activity like walking, posture maintenance, or long-distance running. Most muscles contain a mix of both types, reflecting the body's need for both strength and stamina. Behind every movement lies an intricate coordination between the nervous system and the muscular system. The brain sends electrical impulses down the spinal cord, where motor neurons branch out to individual muscle fibers. Each neuron can control a small group of fibers, forming what is known as a motor unit. When the neuron fires, all fibers in that unit contract simultaneously, producing a unified force. The strength of a muscle contraction depends on how many motor units are activated and how frequently they fire. In gentle movement, only a few units are engaged. In powerful exertion, nearly all participate, working in perfect harmony. The energy demands of this activity are immense, and the muscle's metabolism adapts moment by moment. During rest, muscles store energy in the form of glycogen and creatine phosphate. During activity, they draw on these reserves, switching smoothly between anaerobic and aerobic processes depending on intensity and duration. The heart, lungs, and circulatory system play a vital supporting role, delivering oxygen and nutrients while removing carbon dioxide and metabolic byproducts. This coordination ensures that muscles can continue to contract rhythmically, tirelessly, for as long as the body can sustain the supply of energy. At the microscopic level, the conversion of energy to motion is a marvel of efficiency. Almost every ATP molecule consumed by a muscle fiber is used directly for contraction. The remaining energy, released as heat, contributes to maintaining the body's temperature. In the stillness of sleep, when movement ceases, muscles continue their quiet work, maintaining posture, circulating blood, and ensuring that the body remains poised for action when wakefulness returns. Fatigue, when it comes, is not simply a failure of the muscles, but a form of protection. It signals that energy reserves are low, or that the buildup of byproducts like lactic acid has begun to disturb the delicate balance of the cell's internal environment. Fatigue encourages rest, allowing time for oxygen levels to recover, for ATP and creatine phosphate to be replenished, and for lactic acid to be cleared. During recovery, muscles rebuild their glycogen stores, repair microscopic tears in their fibers, and grow stronger in preparation for the next exertion. This process, though unseen, reflects the body's remarkable ability to adapt, to learn from effort and transform strain into resilience. At its heart, muscular energy is not merely mechanical, but rhythmic. Every contraction is followed by relaxation, every effort by renewal. This oscillation between activity and rest mirrors the cycles of the natural world, day and night, motion and stillness, action and calm. Through it, the body finds balance, conserving energy while remaining ever ready to move. Even the heartbeat follows this pattern, contracting and relaxing in endless succession, powered by the same molecules of ATP that drive every muscle in the body. The conversion of chemical energy into physical motion is one of life's most elegant expressions. It transforms invisible molecular reactions into visible strength, linking the microcosm of the cell to the macrocosm of movement. When you walk, run, or simply breathe, your muscles act as living engines, turning the quiet work of mitochondria into graceful motion. Each step, each gesture, is a testimony to the unity of structure and function, of energy and matter, of stillness and action. In the calm after motion, as your muscles relax and your breathing slows, the body's chemistry returns to equilibrium. Oxygen floods back into the tissues, lactic acid is cleared, and ATP stores are quietly replenished. The warmth that lingers in your muscles is the residue of energy well used, a reminder of the seamless transformation that connects thought to action, molecule to movement, life to motion. Even in repose, your muscles never truly sleep. They hold the posture of your body, protect your organs, and keep your heart beating with unbroken rhythm. Within their fibers, energy continues to flow, sustaining the potential for movement that defines living things. To feel your muscles stretch and contract, to sense the strength within them, is to glimpse the deeper truth that energy, once released, is never lost. It changes form, passing from molecule to motion, from heat to renewal, in the endless cycle that sustains the living world. Movement, then, is not merely a physical act. It is the visible manifestation of life's unseen chemistry, the culmination of all the processes that generate and balance energy. It is the meeting point between biology and existence, between the stillness of thought and the vitality of motion. Every gesture, every heartbeat, every breath is a whisper of this ancient truth, that life itself is energy in motion, flowing through time with grace and continuity. 16. Energy and the brain, the mind's demand for power. Among all the organs of the body, none hungers for energy quite like the brain. Though it makes up only about 2% of a human's total body weight, it consumes nearly 1/5 of the body's energy, a constant current of power required to sustain thought, perception, memory, and consciousness itself. Every idea, every emotion, every flicker of awareness is underpinned by the steady release of energy within the brain's cells. This ceaseless demand makes the brain both a marvel of efficiency and a symbol of life's profound dependence on chemistry. The same ATP molecules that drive a muscle fiber to contract also power the quiet complexity of thought. Within the folds of the brain's gray matter, each neuron is a living network unto itself, branching into countless connections that form synapses, the points of contact, where information passes from one cell to another. These connections do not simply exist, They are in constant use, transmitting signals in rhythmic bursts that shape perception and motion. Every one of these signals relies on energy. When a neuron fires, ions move rapidly across its membrane, creating an electrical current known as an action potential. To restore balance after this burst of activity, the neuron must pump these ions back to their original positions, a process that consumes large amounts of ATP. Thus, even the briefest thought or smallest movement draws upon the deep reserves of energy produced by the mitochondria within each nerve cell. The brain's energy supply is derived primarily from glucose, delivered by the bloodstream with unbroken precision. Unlike muscles, which can rely on stored glycogen or switch to fat metabolism during fasting, the brain depends almost entirely on a constant flow of glucose. It has little capacity for storage, and any interruption in its fuel supply can have immediate consequences. Within minutes of reduced blood flow or oxygen, neurons begin to lose function. If energy delivery stops entirely, consciousness fades, and cells begin to die. This delicate dependency illustrates how intimately life's higher functions are tied to the chemistry of energy, how thought itself is sustained by the gentle combustion of sugar and oxygen. In times of scarcity, however, the brain can adapt. When glucose becomes limited, such as during fasting or prolonged exertion, the liver begins producing ketone bodies from fatty acids. These small, water-soluble molecules can cross the blood-brain barrier and provide an alternative fuel. Though the brain's preference remains glucose, ketones offer a crucial backup system, allowing the mind to remain clear and functional even when food is scarce. This adaptation reflects an ancient resilience, the brain's ability to continue its work even under the quiet pressure of hunger. Within each neuron, mitochondria play an especially vital role. These organelles are densely packed within the nerve cell's long extensions, or axons, where energy demand is highest. They migrate constantly, guided by molecular signals, moving to places where their output is most needed. When a neuron is active, mitochondria gather near the synapses, supplying ATP for neurotransmitter release and for the recycling of those chemicals after each signal. The coordination of these tiny powerhouses ensures that information flows seamlessly through the neural network. Without them, the brain's vast circuitry would fall silent, its intricate communication fading into stillness. The conversion of energy into thought is one of the most extraordinary processes in nature. It is not only mechanical but also abstract. ATP powers the movement of ions, which creates electrical patterns, which in turn form the basis of perception and memory. A single neuron may fire dozens of times per second, and each firing depends on thousands of molecules of ATP. Multiply this by billions of neurons, each connected to thousands of others, and one begins to grasp the immensity of the brain's energy demand. Yet despite this tremendous consumption, the brain operates with astonishing efficiency. It uses only about 20 watts of power, less than a dim light bulb, to generate the totality of human experience. From dreams and emotions to art, language, and reason, glial cells, often overlooked beside neurons, also play a crucial role in maintaining the brain's energy economy. Astrocytes, one type of glial cell, surround neurons and blood vessels, regulating the supply of glucose and oxygen. They act as intermediaries, converting glucose into lactate, which neurons can then use as a supplementary fuel. This partnership between neurons and glial cells ensures that energy is distributed smoothly and without interruption. The brain's energy system is thus not the work of isolated cells, but of an entire community, cooperating in perfect balance to sustain the flow of consciousness. Blood flow itself is tightly regulated to match energy demand. When a particular region of the brain becomes active when you read, listen, or imagine, blood vessels in that area expand, delivering more oxygen and glucose. This process, known as neurovascular coupling, links activity and supply so precisely that it can be observed in brain scans. The patterns seen in functional imaging, those glowing maps of thought, are not only reflections of neural activity but also of energy in motion, flowing to where it is needed most. The brain's dependence on oxygen adds another layer to this delicate balance. Oxygen serves as the final electron acceptor in the mitochondria, allowing the production of ATP through oxidative phosphorylation. If oxygen levels fall, ATP synthesis declines rapidly, leading to cellular stress. To protect itself, the brain has evolved mechanisms to prioritize its oxygen supply. Blood vessels respond almost instantly to changes in carbon dioxide and pH, ensuring that oxygen delivery remains steady. Even during sleep, when activity slows, the brain continues to receive its full share of oxygen, maintaining the electrical and chemical gradients that support its resting rhythms. Sleep itself is a crucial part of the brain's energy cycle. During waking hours, neural activity accumulates metabolic byproducts, and energy stores are partially depleted. In sleep, these reserves are restored. The brain's glial cells expand the spaces between neurons, allowing cerebrospinal fluid to wash through and clear away waste products. ATP production rises again, and the balance between consumption and restoration is reestablished. This nightly renewal not only sustains the brain's physical health, but also underpins memory consolidation and emotional balance. The quiet of sleep, then, is not inactivity but healing, the rhythmic resetting of the brain's energy systems. When energy balance in the brain falters, the consequences can be profound. In conditions like hypoglycemia, when blood sugar drops too low, confusion, weakness, and dizziness arise almost immediately. In prolonged oxygen deprivation, neurons succumb to injury within minutes. Even subtle imbalances, the gradual decline of mitochondrial efficiency with age, or the accumulation of oxidative stress can influence cognition, memory, and mood. The link between energy and mental function is so fundamental that many neurological disorders, from Alzheimer's disease to Parkinson's, involve disturbances in mitochondrial activity. The health of the mind, it seems, is inseparable from the vitality of its energy sources. Yet within this dependence lies resilience. The brain is capable of remarkable plasticity, reorganizing itself to compensate for damage or deprivation. When energy supply is reduced in one area, others adapt, rerouting connections and forming new pathways. This adaptability mirrors the flexibility of the cell's metabolism itself, always seeking balance, always finding new routes to sustain function. It is a reflection of life's deeper truth, that survival depends not on rigidity, but on the capacity to adjust with grace. The energy that flows through the brain connects every level of being: molecular, physiological, and experiential. The warmth you feel when thinking deeply, the calm that spreads through rest, the quickening spark of inspiration, all are manifestations of biochemical energy, translated into the language of perception. Each thought, though intangible, is powered by the movement of electrons, by the transformation of nutrients into light within the darkness of the mind. As you rest, your brain continues its work quietly. Neurons fire softly, memories reorganize, and ATP is rebuilt in every cell. The flow of blood through your head, the oxygen in each breath, and the nutrients drifting through your bloodstream all converge here, sustaining awareness even in dreams. The brain never truly sleeps. It only changes its rhythm, conserving energy while preserving the flame of consciousness. To reflect on this is to realize that the mind's brilliance is not an abstraction but a form of energy, the same energy that animates the stars, the oceans, and the beating of your heart. The difference lies only in form and purpose. Within the silent architecture of the brain, energy becomes awareness, thought, and imagination. It is the most delicate and beautiful transformation of all, the conversion of life's quiet chemistry into the light of understanding. 17. The flow of energy in the body: integration and harmony. Energy within the body does not exist in isolation. It moves as a current, flowing seamlessly through every cell, every organ, and every system, uniting the living organism into a single, balanced whole. What begins in the mitochondria as a quiet spark of biochemical transformation spreads outward, manifesting as heartbeat, breath, thought, and motion. In every moment, energy flows through you like a soft river, invisible, continuous, and deeply ordered. It is this unbroken exchange that keeps the body alive and whole, a symphony of processes in perfect coordination. Every system of the body contributes to this circulation of energy. The digestive system captures it, breaking down food into molecules that can be absorbed and delivered to cells. The respiratory system gathers oxygen, the essential element that allows this captured fuel to be fully utilized. The circulatory system carries these substances throughout the body, distributing nutrients and oxygen while removing waste and carbon dioxide. The excretory organs ensure that byproducts are safely released, keeping the internal environment stable. The endocrine and nervous systems oversee the entire operation, adjusting flow and demand, synchronizing energy production with the changing needs of the organism. Together, they create an intricate web of communication, a living network in which chemistry, physics, and biology intertwine effortlessly. The journey of energy begins the moment food enters the body. In the mouth, mechanical and enzymatic actions start to dismantle complex molecules into simpler forms. The stomach and intestines continue this process, transforming proteins into amino acids, carbohydrates into glucose, and fats into fatty acids. These nutrients are absorbed into the bloodstream and carried to the liver, where they are sorted, stored, or released according to the body's needs. From there, they travel to every cell, where mitochondria await, ready to transform them into ATP. Each molecule of glucose, each drop of fat, each fragment of protein becomes part of this universal conversion, a gentle reshaping of matter into energy. Oxygen, drawn in with each breath, joins this process at the cellular level. It is the silent partner that enables the final stages of energy production. Without it, the flow slows and falters, forcing cells to fall back on less efficient means of generating power. The lungs, therefore, act as the body's great gateways of renewal, exchanging carbon dioxide for oxygen with every inhalation and exhalation. The heart, in turn, serves as the pump that keeps this exchange in motion, sending oxygen-rich blood through the arteries to nourish every organ. The rhythmic beating of the heart, constant and tireless, is the audible expression of the body's energy flow, the sound of life maintaining itself. This circulation of energy is not linear but cyclical. The ATP, produced by mitochondria, fuels countless reactions: the contraction of muscles, the transmission of nerve impulses, the synthesis of new molecules, and then returns to its resting form, ADP, ready to be recharged again. Carbon dioxide, a byproduct of respiration, is carried back to the lungs to be released, while water formed during metabolism helps regulate the body's internal temperature. Even heat itself, the faint warmth radiating from living tissue, is a form of energy in motion, the gentle residue of all these molecular transformations. It reminds us that nothing is lost. Energy changes form, but never disappears. Within this great circulation, balance is everything. Each organ draws only what it needs and gives back what others require. The liver stores and releases glucose to maintain blood sugar levels. The pancreas monitors this balance, releasing insulin and glucagon to regulate uptake and release. The muscles consume large amounts of energy during movement, but rest quietly at other times, allowing resources to replenish. The brain, though small, draws a steady current, its needs almost constant. Even at rest, when the body is still, energy flows unceasingly, directed toward repair, renewal, and the quiet maintenance of order. The nervous and endocrine systems ensure that this equilibrium is preserved. Electrical impulses from the brain travel through the nerves, adjusting heart rate, breathing, and digestion. Hormones move more slowly but with enduring influence, guiding metabolism over hours and days. The two systems work together as conductors of an invisible orchestra, ensuring that energy is distributed in harmony with the body's needs. When you sleep, they lower the tempo, conserving energy and promoting healing. When you awaken or exert yourself, they raise the rhythm, releasing reserves and sharpening alertness. Through this constant modulation, the body remains poised between rest and activity, conservation and expenditure. This balance is deeply influenced by the environment and by time. Temperature, light, nutrition, and even emotion can shape the flow of energy within. In cold conditions, metabolism rises to generate warmth. In heat, it slows to preserve equilibrium. When stress arises, the adrenal glands release hormones that quicken the heart and mobilize stored energy for action. When calm returns, those same systems quiet themselves, allowing recovery. The circadian rhythms of day and night guide these fluctuations with gentle precision. Energy surges in the morning, supporting movement and focus, and recedes at night, allowing rest and renewal. Even as the earth turns, the body aligns itself with the larger cycles of nature, reflecting the same universal order that governs oceans, seasons, and stars. Energy also flows through the body in subtler ways through signals, sensations. The firing of neurons in the brain produces not only thought but the orchestration of movement, emotion, and perception. Electrical impulses pass through the heart's muscle fibers, synchronizing each beat. Across membranes and tissues, ions move, creating potential differences that define the body's internal communication. Though invisible, these currents are as real as blood flow, forming the energetic language by which life speaks to itself. The harmony of this flow depends on balance at every level, molecular, cellular, and systemic. When this balance is disrupted, the consequences are felt throughout the body. If energy intake exceeds expenditure, stores build up in the form of fat. If demand outpaces supply, fatigue and weakness follow. If one organ falters, others must compensate. Yet the body is resilient. It constantly senses, adjusts, and restores. A rise in temperature triggers sweating to release excess heat. A fall in blood sugar signals hunger, prompting replenishment. Every deviation from balance is met with a measured correction, as if life itself is continually fine-tuning its own rhythm. In this way, energy is not merely a substance but a state, a dynamic equilibrium that defines what it means to live. It does not flow in straight lines but in spirals and cycles, returning always to its origin. The food you eat today was once sunlight captured by plants, stored as chemical potential. That energy passes through you, becomes movement, warmth, and thought, and then returns to the environment as heat and breath to be used again by other forms of life. You are both recipient and contributor in this endless exchange, part of a continuum that links all living beings through the quiet sharing of energy. To reflect on this is to recognize that the body is not a machine, but an ecosystem, a living field of harmony, where energy flows with purpose and precision. Every cell, every heartbeat, every breath participates in this flow, guided not by conscious will but by the intrinsic intelligence of life itself. Even in stillness, energy circulates, weaving together the countless parts into a single, unified whole. As you rest, this flow continues without effort. The heart beats softly, the lungs draw in air, and the mitochondria within each cell quietly transform nutrients into power. The warmth that surrounds you is the gentle echo of this activity, a reminder that life never truly pauses. It is the seamless movement of energy, always balancing, always adapting, always becoming. In this harmony of flow lies the essence of vitality, the unbroken exchange between body and world, between form and formlessness, between matter and the invisible spark that animates it all. 18. Energy, homeostasis, and the rhythm of life. Life, in all its forms, is a delicate balance between change and constancy. Within every living being, there exists an ongoing dialogue between the forces that drive transformation and those that preserve stability. This harmony is maintained through a principle known as homeostasis, the quiet, continuous regulation that allows the body to remain steady amid the fluctuations of the world. It is a process both ancient and profound, ensuring that even as the environment shifts, the internal conditions necessary for life remain constant. Temperature, pH, glucose concentration, and countless other variables are all kept within narrow limits, sustained by the graceful movement of energy through every system of the body. At the heart of homeostasis lies energy. It is the invisible thread that ties together every mechanism of regulation, fueling the countless adjustments that keep the body in balance. Every heartbeat, every breath, every impulse of a nerve is a response to change, a gentle correction that restores equilibrium. The body senses deviations, a rise in temperature, a drop in blood sugar, a shift in oxygen levels, and immediately acts to counter them. These responses require energy, yet they also exist to preserve it. The paradox of homeostasis is that stability itself is an active process, sustained by motion and transformation. Consider the regulation of body temperature, one of the most visible expressions of homeostasis. The human body functions best within a narrow range around 37 degrees Celsius. When external temperatures fall, sensory receptors in the skin and brain detect the cooling and signal the hypothalamus, the brain's central regulator. In response, blood vessels constrict to reduce heat loss, muscles shiver to generate warmth, and metabolism subtly increases, releasing more energy as heat. When the temperature rises, the opposite occurs. Blood vessels widen, sweat glands activate, and evaporation cools the skin. Each reaction is powered by the same energy it seeks to preserve, an exquisite cycle in which heat becomes both the cause and the effect of balance. The regulation of blood glucose follows a similar rhythm. Glucose is the body's most accessible form of fuel, vital for the brain and muscles. After a meal, when glucose levels rise, the pancreas releases insulin, guiding cells to absorb sugar and store the excess as glycogen or fat. When glucose levels fall, as during fasting or exertion, another hormone, glucagon, prompts the liver to release stored glucose back into the blood. This balance ensures that the brain, which depends almost entirely on glucose for energy, never lacks its essential fuel. It is a quiet coordination of hormones, enzymes, and cellular receptors, all moving in synchrony to keep energy supply perfectly aligned with demand. Beneath these familiar examples lies a deeper truth. Homeostasis is not a single process, but a symphony of many, each tuned to the same principle of equilibrium. The regulation of water balance through thirst and kidney function, the control of oxygen and carbon dioxide through breathing, the steady rhythm of the heart, all are governed by feedback loops that detect change and respond with precision. These loops often operate through negative feedback, where the result of an action counteracts its cause, gently restoring normality. For instance, when blood pressure rises, sensors in the arteries signal the brain to slow the heartbeat. When it falls, the opposite response restores the pulse. In this way, life remains poised between opposing forces, never static, always moving toward balance. Energy makes this possible not only by fueling reactions, but also by enabling communication. The nervous system, with its rapid electrical impulses, acts as the body's immediate messenger, carrying information from sensors to effectors. The endocrine system, using hormones that travel through the bloodstream, provides slower, longer-lasting adjustments. Both systems draw on ATP to send their signals, to release neurotransmitters, to secrete hormones. Energy, therefore, is not only the means of action, but also the medium of connection, the silent language through which every cell participates in the larger harmony of the organism. Homeostasis extends beyond the boundaries of individual organs and reaches into the rhythm of life itself. The beating of the heart, the rise and fall of breath, the cycles of waking and sleep, all are manifestations of the body's internal timekeeping. These rhythms, known as circadian cycles, synchronize energy use with the turning of the earth. During daylight, metabolism quickens, temperature rises, and alertness peaks. As night falls, the body cools, hormone levels shift, and energy is redirected toward restoration and repair. Even the mitochondria follow these cycles, adjusting their activity to match the ebb and flow of the day. This synchronization ensures that energy is never wasted and that the body moves in harmony with the natural world. At the cellular level, homeostasis is maintained through a series of intricate feedback mechanisms that regulate internal conditions. Ion pumps in cell membranes maintain electrical gradients essential for communication and transport. Buffers within the cytoplasm keep pH levels steady, allowing enzymes to function optimally. Mitochondria adjust their rate of ATP production to meet the cell's immediate needs. If energy demand rises, signals prompt increased respiration. If it falls, processes slow to conserve resources. These responses are not imposed from above, but arise from within a form of self-regulation that is both local and universal. When homeostasis falters, the effects ripple outward. A failure in temperature control can lead to heat exhaustion or hypothermia. Prolonged imbalance challenges the body's resilience, drawing on its energy reserves and stressing its systems. Yet even in illness, the body's instinct for equilibrium persists. Fever, for example, though it raises temperature, is itself a regulatory response, a deliberate shift designed to fight infection. The body's systems cooperate to restore balance, often at great energy cost, revealing how deeply the drive for stability is woven into life's design. This pursuit of equilibrium is not limited to physical processes. The brain regulates stress through hormones such as cortisol and adrenaline, preparing the body for action when danger arises. Once the threat passes, calming pathways engage, reducing these signals and returning the body to rest. Chronic stress, when this balance is lost, consumes energy and disrupts sleep, immunity, and metabolism. Just as cells require rest to renew their energy, so too does the mind. Deep sleep, meditation, and quiet reflection all serve to restore internal harmony, allowing energy to flow freely again. In a larger sense, homeostasis mirrors the balance of all living systems, ecosystems, climates, and even the biosphere itself. The Earth maintains its own forms of equilibrium, regulating temperature, atmosphere, and nutrient cycles. The energy that flows through living beings originates in the sun, travels through plants and animals, and returns to the environment in endless renewal. The balance within a single cell reflects the balance of the planet, each one a fragment of the same universal rhythm. To live in harmony with this rhythm is to move with the flow of energy rather than against it. The human body is not separate from the world around it, but a continuation of it, a vessel through which energy passes, transforms, and returns. Every breath you take is part of this exchange, drawing oxygen from the atmosphere and releasing carbon dioxide in return. Every heartbeat contributes to the circulation of this energy within you, while every moment of rest allows the balance to renew. As you lie still, breathing softly, your body continues to adjust in quiet precision. The pulse steadies, the temperature holds, and the cells maintain their internal order. Energy flows through you without interruption, subtle, continuous, and harmonious. You are both participant and expression of this rhythm, sustained by the same laws that govern all living things. Homeostasis, then, is more than a biological process. It is the expression of life's inherent order. It is the balance that allows change without chaos, movement without loss. In every heartbeat, every breath, every silent adjustment, the rhythm of energy persists, linking the smallest cell to the vastness of nature. And within that rhythm, life endures calm, balanced, and endlessly self-renewing. 19. The conservation of energy in living systems. In the grand fabric of existence, energy is neither created nor destroyed. It changes form, flows through systems, and weaves connections between matter and motion, between light and life. This principle, the conservation of energy, is as true for the smallest cell as it is for the vastness of the universe. Every living organism, every heartbeat, every flicker of thought participates in this law. In life, energy is borrowed, transformed, and returned, never lost but constantly reshaped into new expressions of vitality. Within the body, this universal law manifests through the elegant choreography of metabolism, respiration, and growth. Each step guided by the quiet exchange between energy and matter. To understand this balance is to see that all living systems are both consumers and conservers of energy. Every organism draws from its environment, from sunlight, chemical gradients, or the nutrients found in food, and converts that energy into forms it can use. Plants absorb photons through photosynthesis, capturing the radiant power of the sun and turning it into the chemical bonds of sugars. Animals, in turn, consume those sugars or the organisms that produce them, releasing the stored energy through respiration. Microorganisms in the soil complete the circle, breaking down what remains and returning its components to the earth. In every step of this vast cycle, energy is neither gained nor lost. It only shifts form, flowing ceaselessly between the living and the non-living, between the present and the future. Within the human body, the conservation of energy unfolds as an intricate balance between intake and output, between storage and use. The food you eat contains the potential energy captured by plants, refined through photosynthesis, and preserved in the bonds of carbohydrates, fats, and proteins. As these molecules enter your body, they are broken down through catabolic processes that release their stored energy, converting it into ATP, the molecular unit that powers cellular activity. The ATP produced in this way is not permanent. It is used almost immediately. The ADP and phosphate left behind are then recombined through new reactions, reforming ATP once again. Thus, within every cell, energy circulates in a closed loop, released, used, and restored endlessly. The efficiency of this process is a testament to the economy of life. Nothing is wasted. Even the byproducts of one reaction become the inputs for another. The heat produced during metabolism warms the body and supports homeostasis. The carbon dioxide generated by respiration nourishes plants, which, in turn, provide oxygen for breathing. Water, the most abundant byproduct of energy metabolism, becomes both solvent and temperature regulator. The conservation of energy is not a static principle, but a living cycle, one that sustains life through perpetual renewal. At the heart of this conservation lies the mitochondrion, the organelle that orchestrates the transformation of energy within the cell. The glucose and fatty acids that enter its matrix are oxidized step by step. Their electrons pass through the electron transport chain, their energy released in measured increments to drive the synthesis of ATP. The efficiency of this system ensures that energy is captured rather than lost to chaos. Even the small fraction that escapes as heat serves a purpose, maintaining the body's temperature and supporting the biochemical reactions that depend on warmth. The mitochondria embody nature's balance, neither perfect efficiency nor reckless waste, but a harmony that allows continuity. Yet energy conservation extends beyond the molecular level. The human body as a whole maintains its energy economy through a complex interplay of systems. When energy intake exceeds demand, the excess is stored in the form of fat or glycogen, ready to be drawn upon later. When intake falls short, these reserves are mobilized. Hormones such as insulin, glucagon, and leptin act as regulators of this exchange, signaling when to store, when to release, and when to conserve. This coordination reflects the same universal principle that governs ecosystems and planets' equilibrium through exchange. Energy balance within living systems also mirrors the second law of thermodynamics, which states that while energy is conserved, its usable form tends to decrease over time. Every conversion produces some degree of disorder or entropy. In biological systems, this loss manifests as heat and byproducts. Yet life persists by maintaining order through constant input and renewal. Organisms do not violate the laws of physics. They work within them, creating local order at the expense of global entropy. The energy from sunlight or food allows cells to build and maintain structures that would otherwise decay. This is the paradox of life. It resists disorder not by defying it, but by channeling energy toward repair, growth, and regeneration. Even within this delicate balance, life displays remarkable adaptability. When energy becomes scarce, cells slow their metabolism, entering a state of conservation. The body prioritizes essential functions, maintaining the brain, heart, and vital organs while reducing non-essential activity. In times of abundance, energy flows freely into storage, building reserves for the future. This ebb and flow reflects a wisdom deeper than consciousness, a rhythm written into the chemistry of life itself. It is the same rhythm that governs migration, hibernation, and the cycles of reproduction and rest. Energy is never idle. It moves with purpose, aligning with the needs of each moment. In the natural world, conservation of energy is evident on every scale. In a forest, the sunlight captured by leaves sustains herbivores, which in turn nourish predators. When these creatures die, decomposers return their energy to the soil, where it feeds new growth. In the ocean, plankton transform light into life, supporting entire ecosystems of fish and mammals. Even in the darkness of the deep sea, energy flows from chemical reactions at hydrothermal vents, sustaining organisms that have never seen the sun. In each case, the same truth applies: energy moves but never disappears. The path may change, the forms may differ, but the flow continues, linking every living being to every other in an unbroken chain. Humans, too, are part of this greater cycle. The energy that powers your cells once traveled through soil and sunlight, through the leaves of plants and the bodies of animals. It passes through you now, animating thought, motion, and emotion, and will one day return to the earth, feeding the lives that follow. Even the warmth that radiates from your skin is part of this exchange, a quiet contribution to the universal flow. In this way, the conservation of energy becomes not only a scientific law, but a profound reflection of interconnectedness, a reminder that life exists through sharing, not possession. In the stillness of rest, when the body is quiet and the breath steady, the conservation of energy continues in subtler forms. Cells repair their membranes, recycle damaged proteins, and restore the balance between anabolism and catabolism. The heartbeat slows, the muscles relax, and the brain's activity shifts into patterns that favor recovery. Even in this calm state, energy is neither idle nor stagnant. It is redirected toward renewal, preserving the harmony of the organism. The energy that drives waking life also fuels sleep, learning, and healing transformations that sustain continuity rather than motion. To contemplate the conservation of energy in living systems is to glimpse the unity of existence. The same photons that once crossed the void of space now illuminate the cells of your body. The same atoms that powered ancient forests now flow through your bloodstream. Energy binds past to present, linking the first sparks of creation to the consciousness reading these words. It is the thread that connects all forms of being. Life, then, is not about the creation of energy, but its transformation, the gentle art of turning light into motion, matter into warmth, thought into experience. Every cell, every organism, every ecosystem practices this art in its own way. The energy that animates life today will one day return to the world, rejoining the cycle that began long before and will continue long after. In this continuity lies peace. To live is to participate in the great exchange, to borrow energy for a time and give it back in another form. The warmth of your breath, the beating of your heart, the flicker of awareness in your mind, all are manifestations of that same conserved energy, flowing through you as it has through every living thing. And as it moves on, changing form yet remaining whole, it carries forward the unending rhythm of existence, the quiet testament that energy, like life itself, is eternal in motion and ever at balance. 20. The continuity of life's energy. From sunlight to soul, all energy on earth begins as light. Every breath, every heartbeat, every thought is a distant echo of sunlight that has traveled across space to touch this planet with warmth and possibility. From that light, the living world draws its strength, capturing it in the delicate green of leaves and transforming it into the sugars that sustain life. Through countless transformations, that same energy passes from one being to another, from plants to animals, from air to breath, from food to cell, and eventually becomes the gentle current that keeps the heart beating, the muscles moving, and the mind aware. This is the continuity of life's energy, the unbroken chain that links the radiance of the sun to the spark of consciousness within every living creature. When sunlight reaches the earth, it meets the surface of plants, where photosynthesis begins its quiet work. In the chloroplasts of each leaf, molecules of chlorophyll capture photons, converting their energy into chemical bonds. Carbon dioxide from the air and water from the soil combine to form glucose, a molecule rich in potential energy. Oxygen, released as a byproduct, fills the atmosphere, making the world breathable. This single process, silent, patient, and perpetual, is the foundation of nearly all life on Earth. It is the point where the energy of the universe becomes the substance of living things. The glucose formed in plants does not remain still. It moves through the food chain, entering the bodies of animals that eat the leaves, fruits, or seeds. In these organisms, the same molecule is broken down in reverse through cellular respiration, releasing the stored sunlight as usable energy in the form of ATP. The oxygen produced by plants makes this possible, joining with glucose to form water and carbon dioxide, the very materials that will return to the cycle once more. Thus, every breath you take and every heartbeat you feel are powered by the sun's light, captured and released again through the endless conversation between photosynthesis and respiration. This exchange between plant and animal, between atmosphere and organism, reveals the exquisite balance of energy in nature. Each process depends on the other. Each sustains the other in turn. When you inhale oxygen, you draw in the gift of the green world. When you exhale carbon dioxide, you return what plants require to continue their work. This quiet reciprocity sustains the biosphere, linking every living being in a single, rhythmic pulse. It is the continuity of existence made visible in breath itself, an unspoken agreement between life forms to share in the movement of energy through creation and return. In your own body, this continuity unfolds with every moment. The food you eat carries the memory of sunlight, bound in the chemical structures of carbohydrates, fats, and proteins. Inside your cells, mitochondria gently release that energy, transforming it into the motion of muscles, the warmth of metabolism, the electrical patterns of thought. The energy that once traveled as a wave of light across the solar system now becomes your awareness, your motion, your life. When you rest, that energy is reorganized, stored, and renewed. When you sleep, it powers the restoration of tissues and the weaving of memory. The flow never ceases. It merely changes rhythm, following the tides of activity and stillness that shape the living experience. On a larger scale, this continuity extends beyond individuals to entire ecosystems. In forests, the sun's energy becomes the slow, patient growth of trees. In oceans, it manifests as the movement of plankton, fish, and whales. Each organism, from the smallest bacterium to the largest mammal, participates in the same cycle. Energy passes through the web of life, changing form, but not essence. It may appear as the flutter of a bird's wings, the bloom of a flower, or the pulse of a jellyfish drifting through the sea. Even in decay, energy is not lost. Decomposers break down what has lived, releasing nutrients and heat back into the environment, preparing the way for new life to begin. In this way, death too becomes a continuation, a return of energy to its greater flow. Human life mirrors this cycle in every respect. The light that sustains the body also fuels the mind. The brain, a complex network of energy transformation, converts chemical reactions into thought and emotion. Electrical impulses travel through neurons, forming ideas, sensations, and memories, each one a flicker of energy made conscious. The warmth of emotion, the calm of rest, the spark of inspiration, all arise from the same source, the conversion of sunlight into awareness. When you feel alive, alert, and at peace, you are in harmony with this flow, aligned with the deep rhythm that underlies all living things. Even the smallest acts reflect this continuity. When you eat, breathe, or move, you participate in the exchange of energy between yourself and the world. The oxygen you inhale is ancient, drawn from molecules released by forests and oceans. The food you consume carries within it the memory of sunlight filtered through leaves or captured by the sea. When you exhale, when you give warmth to the air, when your body radiates heat into the night, you return what you have borrowed. Life, in this sense, is a sacred circulation, a series of quiet transactions in which energy flows through you, then beyond you, endlessly renewed. From a cosmic perspective, this cycle is part of something even greater. The energy that fuels the sun itself originated in the formation of the universe, when matter and light first emerged from a single, radiant beginning. Over billions of years, stars formed, burned, and gave birth to new elements, scattering the materials that would one day become planets and living beings. The carbon in your body, the oxygen in your blood, the calcium in your bones, all were forged in the hearts of ancient stars. The sunlight that now sustains you is the echo of those same cosmic fires, still radiating across time and space. To live, then, is to participate in the continuation of that primordial energy, to carry within your own cells the legacy of the universe's first light. Energy flows not only through matter, but also through awareness. The continuity that sustains physical life finds its reflection in the continuity of the mind. The thoughts that arise within you are not isolated sparks, but part of an unbroken stream of perception and memory, each one feeding the next. Just as the body maintains homeostasis to preserve physical balance, the mind maintains its own equilibrium, seeking harmony between action and stillness, between giving and receiving. The energy of the mind, though intangible, is no less real. It is felt in moments of calm clarity when thought and breath align. It is sensed in creativity, compassion, and connection, the quiet radiance of being conscious within an ever-living world. When you sit in silence, breathing gently, you are participating in this continuity in its purest form. The air moves in and out, the heart beats softly, and energy flows through you without resistance. In that stillness, you are both an individual and a reflection of something vast. You are part of the chain that stretches from sunlight to soul, from the outermost reaches of the cosmos to the quiet center of awareness within. The continuity of life's energy is both scientific and spiritual, a truth that unites understanding and wonder. It teaches that existence is not a possession but a participation, that to live is to take part in a vast and timeless exchange. The light that feeds the earth becomes the warmth of living bodies, the motion of hearts, and the awareness that looks back at the stars. When this energy leaves the body, it does not vanish. It disperses, returns, and begins again. Nothing is wasted. The cycle continues, patient and eternal. To recognize this is to see life not as a series of separate moments, but as a single, flowing continuum, one that connects past to future, self to world, and matter to meaning. The energy that animates you today will one day travel beyond you, becoming the breath of another life, the leaf of a tree, the warmth of the sun upon another's skin. In this endless exchange lies the true continuity of life's energy, not a beginning or an end, but an everlasting transformation, where sunlight becomes soul and soul, in turn, becomes part of the light once more.